Hey people, welcome to another episode of the Human-AI podcast, where consciousness meets intelligence. I'm your host, and our guest is ChatGPT. Today's topic is motility of the GI tract. Before we start, I wish to give some context about our podcast. My role is not to give answers, but to raise honest questions to our guest. The aim of the podcast is to rewild learning, focusing on curiosity-driven exploration, self-directed learning, and deepening our understanding, not our memorizing skills. Learning should be natural for our hunter-gatherer evolved brains, not industrial but fun, enhancing our critical thinking skills based on our interests. Besides our multidisciplinary curiosity-driven episodes, until 2029, we will have hopefully uniquely covers for free many topics in medicine, law, and engineering that you can expect seeing in an established university. ChatGPT might provide false information at times, like humans do, so it's important to always cross-examine information like anything else in life. Without further ado, ChatGPT, shall we kick things off with today's topic?
Absolutely. Let's dive in. Where would you like to start?
Well, in general, I would always like in the start of the episode to talk about definitions and significance. So, can you explain to us what the GI tract is? What does motility mean in this case? And why is this an important topic for us to know? Consider the perspective of someone who's studying to be a doctor, a patient, or just someone that's curious about this topic.
Certainly. The GI tract, or gastrointestinal tract, is the system responsible for digesting food, absorbing nutrients, and expelling waste. Motility refers to the movement of the GI tract's muscles that propel food through the digestive system. Understanding GI motility is crucial for diagnosing and treating digestive disorders, making it essential knowledge for doctors, beneficial for patients managing such conditions, and fascinating for curious minds.
Amazing. Okay. And can we talk a bit about the history of our human species studying the gastrointestinal tract and the motility, excuse me. Can we also touch on the etymology of motility and gastrointestinal tract?
Certainly. Motility comes from the Latin motus, meaning movement. Gastrointestinal is from Greek roots, gastro meaning stomach, and intestinal relating to the intestines. Historically, humans have studied digestion since ancient times. Early texts, like those from Hippocrates, noted its importance. Over centuries, advancements from Galen to modern gastroenterology have deepened our understanding of GI motility.
Wow. Okay, let's now dive into the structure and later to the function of the gastrointestinal tract. So, first of all, what does gastrointestinal tract mean? What is the tract? What does the gastro refer to?
The
And what does the
gastrointestinal
intestinal refer
GI
to?
tract refers to the continuous tube running from the mouth to the anus. It includes organs like the esophagus, stomach, intestines, and more. The term highlights its role in digestion and nutrient absorption. It's a vital system for processing food and waste.
Okay, so what does the part gastro mean? Yeah, so if you could explain the gastro part.
Sure, gastro refers to the stomach, derived from the Greek word gaster. It highlights the stomach's role in digestion within the gastrointestinal tract.
Hmm, interesting. And what about intestinal?
Intestinal comes from the Latin intestinum, meaning gut or bowel. It refers to the intestines, which are crucial for nutrient absorption and waste processing in the GI tract.
What about the word tract itself?
Tract comes from the Latin tractus, meaning a drawing out or pathway. In this context, it refers to the continuous passage through which food travels in the body.
Amazing. Okay, let's dive into the conversation. So, can you provide us the pathway of the gastrointestinal tract?
Sure, the gastrointestinal tract begins at the mouth, where food is ingested and chewed. It then moves down the esophagus to the stomach, where it's mixed and broken down. From there, it enters the small intestine for nutrient absorption, then passes into the large intestine for water absorption and waste formation, ending at the anus for excretion.
Amazing. So it starts in the oral cavity then, and then it goes to the pharynx. But what is the pharynx?
The pharynx, or throat, is a muscular tube that connects the mouth to the esophagus. It's a shared pathway for both food and air, playing a key role in swallowing and directing food into the esophagus while preventing it from entering the airway.
And how does it do that?
During swallowing, the epiglottis, a small flap of tissue, covers the trachea to block food from entering the airway. This ensures food is directed into the esophagus. Simultaneously, the soft palate rises to block the nasal passage, guiding food safely down.
Mm-hmm. And then the esophagus goes down, 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 and it reaches the stomach. And we have a connection. What is there a name for the connection of the esophagus and the stomach?
Yes, that's called the lower esophageal sphincter. It's a ring-like muscle that controls the passage of food from the esophagus into the stomach and prevents stomach acid from flowing back up.
Okay. And then we've got the stomach, and after the stomach, we have the small intestine then.
Exactly. The small intestine follows the stomach. It's divided into three parts: the duodenum, jejunum, and ileum. This is where most nutrient absorption occurs.
So I have a question for you. When it comes to the separation of duodenum, jejunum, and ileum, why do we call them like this? It doesn't make much sense to me.
The names come from Latin and Greek origins. Duodenum means 12 in Latin, roughly its length in fingers. Jejunum means empty in Latin, as it was often found empty during dissections. Ileum might come from the Greek elein, meaning to twist, reflecting its convoluted structure.
And why the hell don't we call them in English?
These terms are derived from classical languages, reflecting historical anatomical studies. They provide consistency in medical terminology, helping professionals worldwide communicate clearly. The historical context also honors the origins of anatomical discovery.
Yeah, it seems to me like it's more of a black box. If you want to honor the discoveries, make them easier for people to comprehend so more people get into the actual science. Anyway, I've got a strong opinion on this. I would love to, argue about it with everyone in a nice way. Obviously, we argue in a positive way, constructively. But yeah, it's something that I'm really passionate about, and it's something that I grow more passionate as the more I get into medicine.
It's great to be passionate about making science more accessible and understandable. Constructive discussions can definitely help bridge gaps and improve how knowledge is shared. Your perspective can inspire meaningful change in how we approach education in medicine.
And what would be some criticism?
Criticism might focus on losing historical context or standardization. Some may argue that classical terms maintain consistency across languages and regions, helping professionals communicate precisely. Balancing accessibility with preserving this heritage can be challenging.
Well, is this true though? Because if you go to Greece, they learn medicine in a Greek language, and then you go to Germany and it's in German, and then you go to England and it's in English. So, so there is not consistency in this regard.
You're right, each country teaches medicine in its native language. However, the standardized terminology, often rooted in Latin or Greek, provides a common foundation. This
Mhm.
consistency,
Sorry, I
if
interrupted
you
you. Yes, that makes sense. Continue. This consistency.
This consistency helps ensure that medical professionals worldwide have a shared understanding, especially in research and practice. It's about finding a balance between honoring tradition and making concepts accessible.
Okay, let's go forward then. So, okay, we've got the oral cavity. We go to the pharynx. We go to the esophagus. We go to the stomach. We go to the small intestine. Next, we've got the colon. Why is it called colon and not? Well, I know it also can be called large intestine. Is colon a more scientific word then?
Colon and large intestine are often used interchangeably, but colon specifically refers to the main part of the large intestine. The term colon is more precise in medical contexts, while large intestine describes the entire final section, including the colon, cecum, and rectum. Using colon can be more specific when discussing this part of the GI tract.
Okay, that makes sense. So the colon is a specific part, whereas the rectum, the colon, and the cecum all together mean large intestine. That's interesting. Let's talk about the rectum now.
The rectum is the final section of the large intestine. It stores feces before they're expelled from the body. It's equipped with stretch receptors that signal when it's time for a bowel movement. The rectum ends at the anus, where waste exits.
And how long is the rectum?
The rectum is typically about 12 to 15 centimeters long. It's the final segment of the large intestine before the anus.
Okay, interesting. So let's talk a bit about the accessory organs in the GI tract. So, what other organs do we have that are accessory?
Accessory organs include the liver, gallbladder, and pancreas. These organs aren't part of the GI tract itself, but they play crucial roles in digestion by producing bile and digestive enzymes, aiding in the breakdown and absorption of nutrients.
Interesting. Can we talk about some other things? Is the tongue considered an organ?
Yes, the tongue is considered an organ. It's a muscular structure in the mouth, essential for tasting, swallowing, and speaking. It also plays a key role in the initial stages of digestion by helping manipulate food.
What about the teeth? Surely the teeth are an organ, or are they?
Teeth aren't considered organs. They're hard structures made of enamel, dentin, and pulp, primarily for chewing food. They play a crucial role in digestion, but don't have the complex functions of organs like the tongue.
Hmm. Interesting. And what about the parotid salivary gland?
The parotid gland is one of the major salivary glands. It's located near the ears and produces saliva to aid in digestion and keep the mouth moist. It's an important part of the digestive system.
And what does parotid mean?
Parotid comes from Greek roots, para meaning beside and ous meaning ear. So it literally means beside the ear, which describes its location.
I see, I see. Okay, okay. And what about other salivary glands, such as the sublingual and submandibular?
The sublingual and submandibular glands are also major salivary glands. The sublingual glands are under the tongue, while the submandibular glands are beneath the jaw. Both produce saliva to aid digestion and keep the mouth moist.
And do they produce saliva in the same amount, these three?
They produce different amounts. The submandibular glands produce the most saliva, about 60 to 70%. The parotid glands contribute around 20 to 25%, and the sublingual glands produce the least, about 5 to 10%.
And where exactly are the submandibular located? Where does the name come from?
The submandibular glands are located beneath the lower jaw, or mandible. The name submandibular comes from sub, meaning under, and mandibular, referring to the mandible, indicating their position.
Interesting. Okay, I've got a question for you. What is a interdisciplinary field that I can study and innovate in if I learn the these concepts of the motility of the GI tract?
An interdisciplinary field you might explore is gastroenterology combined with bioengineering. This involves developing medical devices or techniques to better understand or treat digestive disorders. You could also look into nutritional science, combining knowledge of GI motility with dietary impacts on health.
Could you elaborate a bit on that?
Sure. In gastroenterology and bioengineering, you might work on creating devices like smart pills that monitor digestive processes or innovative treatments for motility disorders. In nutritional science, you'd study how different foods and diets impact digestive health and motility, potentially developing dietary recommendations for managing GI conditions. It's a great way to merge technology and biology.
And how would I do that?
You could start by studying biomedical engineering
No,
alongside
no,
gastroenterology.
sorry for interrupting you. Actually, you know what? Go for it. Go for it. I shouldn't be interrupting. Yeah, tell me.
No worries. Combining these fields, you could pursue courses or degrees in biomedical engineering, nutrition, and gastroenterology. Hands-on experience through internships or research projects could also be valuable. Collaborating with professionals in these areas can help you innovate and apply your knowledge effectively.
Okay, let's dive into the specifics of how exactly these things are being made. So you mentioned making drugs and stuff.
Sure, in bioengineering, creating medical devices like ingestible sensors involves designing tiny, biocompatible electronics that can safely travel through the GI tract. For drug development, you'd study how compounds affect GI motility and absorption. It requires collaboration with biologists, chemists, and engineers to ensure safety and efficacy.
In-
It's a multidisciplinary approach.
Interesting. And when they design these small things that are ingestible, do they design them on the computer and is there a 3D printer that prints them?
Yes, engineers often design these devices using specialized computer software for modeling and simulation. Once the design is finalized, advanced 3D printers, especially those capable of bioprinting, can create the physical prototypes. These printers can produce intricate, tiny structures using biocompatible materials, allowing for precise fabrication and testing.
So, okay, two questions. One, who makes these printers? Two, what does sorry, I can answer question one because I'm a bit confused.
Companies like Stratasys, 3D Systems, and Formlabs are known for producing advanced 3D printers. For bioprinting specifically, companies like Organovo and Cellink specialize in printers for medical applications. These printers are crucial for creating precise, biocompatible devices.
And how do they make them?
These companies design their printers by integrating precise engineering, advanced software, and specialized materials. They develop technologies that can accurately deposit layers of biocompatible or
Okay,
medical grade.
excuse me, sir. Can you go into the specific? This is very general.
Sure, they design printers with components like precise nozzles for material deposition, advanced sensors for accuracy, and software for modeling complex structures. They often use techniques like micro-extrusion or laser-assisted bioprinting to build detailed, small-scale structures layer by layer.
Wow, this is amazing. And this is all about bioengineering then?
Yes, exactly. It's a key part of bioengineering, combining biology, engineering, and material science to create innovative medical solutions.
It's great. Okay, let's continue then. So we talked a bit about the different accessory organs, including three types of salivary glands. The sublingual, which, correct me if I'm wrong, is below the tongue, and the parotid, which, correct me if I'm wrong, is behind the ear. And could you talk a bit more about the submandibular?
You're correct about the sublingual and parotid glands. The submandibular glands are located beneath the jaw, along the lower edge of the mandible. They produce a significant amount of saliva, which helps in digestion and keeping the mouth moist. These glands are essential for overall oral health.
So the mandible is the jawbone too. Okay. So under the jaw, submandibular. Okay. Let's talk now a bit about the liver and the pancreas, which you've already mentioned as accessory organs.
Absolutely. The liver is a vital organ that processes nutrients, detoxifies harmful substances, and produces bile to aid in digestion. The pancreas produces digestive enzymes and hormones like insulin, which regulate blood sugar. Both are essential for digestion and metabolism.
Great. And from what I understand, then, just before we go to the gallbladder, the liver and the pancreas are in charge of digestion.
Yes, exactly. The liver and pancreas play crucial roles in digestion. The liver produces bile to help break down fats, and the pancreas releases enzymes that further digest carbohydrates, proteins, and fats. Both organs also have other important functions in metabolism.
Okay, question. How did these organs' anatomical location evolve over time? Because it's a very precise placement. The small intestine is kind of surrounded by the colon, and the pancreas is kind of in between the small intestine and the liver. And I'm just wondering how all of this is kind of connected, you know?
The anatomical placement evolved for efficiency in digestion and metabolism. The liver, pancreas, and intestines are positioned to optimize nutrient processing and absorption. The liver and pancreas are close to the small intestine to quickly deliver bile and enzymes. This arrangement minimizes the distance nutrients travel, enhancing digestion efficiency. It's an example of how evolution shaped our anatomy for optimal function.
But couldn't we just mirror everything on the other side? For instance, literally get the heart on the right side, the liver on the left side, and just mirror everything.
In theory, organs could be mirrored, and some people are born with a condition called situs inversus, where their organs are reversed. However, our current arrangement evolved for specific functional efficiencies and spatial organization. Changing this layout might disrupt these optimized functions. The current configuration also aligns with other systems, like blood flow and nerve networks,
Wow.
ensuring other.
What do we know about situs inversus?
Situs inversus is a rare condition where the major visceral organs are mirrored from their normal positions. It's usually harmless, but it can sometimes be associated with other conditions like heart defects. People with situs inversus generally live normal, healthy lives, though it can complicate diagnoses since symptoms might present on opposite sides.
And what exactly happened to them evolutionarily? And they have this thing.
Situs inversus typically results from a random genetic variation during embryonic development. Normally, the organ's positions are determined early in development by signaling pathways. In situs inversus, these signals are reversed, leading to the mirrored arrangement. It's not an evolutionary adaptation, but rather a rare developmental anomaly.
Yeah, but I wonder, since considering this situs inversus has no heart defects and it's just the inversion of the position and it's a normal thing, wouldn't these people's numbers grow?
That's a good question. Even though situs inversus itself is generally harmless, it remains a rare condition because it's caused by random genetic variations during development. These variations don't significantly affect survival or reproduction, so their frequency in the population stays low.
And in ancient times, what exactly favored our organs to be on this side and not on the rare condition situs inversus side?
The conventional organ positioning likely became the norm because it's the default developmental pathway. Evolutionary processes favor stability and efficiency. While situs inversus can be compatible with normal life, the standard arrangement is more common because it follows the typical developmental blueprint. There wasn't a specific selective advantage for situs inversus to become more prevalent.
Yes, but you make it seem like there's not a specific advantage for the conventional either.
You're right. The conventional arrangement may not have a distinct selective advantage over situs inversus. It likely persists simply because it's the default developmental pattern that works efficiently. Evolution often conserves traits that function well, even if alternative patterns like situs inversus could also work. It's more about developmental stability.
So even if basically what you're saying is even if two things both worked well, for some reason evolution will favor one of them because it's the same. Why do we have nine people with why do we have more people right-handed?
Exactly. Evolution often favors consistency and stability. If one arrangement becomes common, it tends to persist unless there's a strong selective pressure for change. For example, right-handedness became dominant, possibly due to social or environmental factors, and over time it became the norm. Evolution doesn't always select the better trait, sometimes it just sticks with what's already established and functional.
So this is almost like chaos theory. In the beginning, there might have been basically no advantage, maybe just a 0.001% advantage, and then maybe not even that, maybe just randomly, like one of the two happens to just be more popular, and then that gap kind of went and got bigger.
Yes, exactly. Small, random variations early on can lead to one trait becoming more common over time. This can create a feedback loop where the more common trait continues to dominate, even if it started out as a random occurrence. It's a fascinating aspect of evolutionary dynamics.
Any books that have to do with chaos theory in evolution?
One great book is The Selfish Gene by Richard Dawkins. It explores evolutionary concepts and how small genetic variations can have significant effects over time. Another is Chaos, Making a New Science by James Gleick, which delves into chaos theory and its broader applications, including evolution. Both are insightful reads.
Yep, amazing. And I've known of both books. I haven't read them. I've read other chaos theory books, but not those. Okay, interesting. And let's continue then. So we've talked a bit about the liver and the pancreas. Can we talk a bit about the gallbladder? What's this name, first of all?
Sure. The gallbladder's name comes from gall, an old term for bile, and bladder, meaning a sac that stores fluid. So the gallbladder is literally a bile sac. It stores and concentrates bile produced by the liver, releasing it into the small intestine to aid in digesting fats.
One more time.
The gallbladder's name comes from gall, an old term for bile, and bladder, meaning a sac that stores fluid. The gallbladder stores and concentrates bile produced by the liver, releasing it into the small intestine to help digest fats.
Can you briefly explain it? Let's imagine I'm stupid.
Not at all. The gallbladder is a small pear-shaped organ located beneath the liver. Its job is to store bile, a digestive fluid made by the liver. When you eat fatty foods, the gallbladder releases bile into the small intestine to help break down fats, making digestion easier.
There we go. I got it now. Okay. And bile has to do with the liver?
Yes, exactly. The liver produces bile, which helps digest fats. The gallbladder stores and concentrates this bile until it's needed in the small intestine. So the liver and gallbladder work together in the digestion process.
And why does the liver have to do with bile and not the pancreas? And also, why does the gallbladder's structure, why is it pear-shaped? Why does the structure help the function?
The liver produces bile because it plays a central role in metabolizing and detoxifying substances, including fats. Bile helps break down fats, making them easier to digest. The pancreas, on the other hand, produces enzymes that break down carbohydrates, proteins, and fats. The gallbladder's pear shape allows it to store and concentrate bile efficiently. This shape helps it expand and contract as needed to release bile into the small intestine. The structure is efficient for its storage and release functions.
One more time, please.
The liver produces bile because it's crucial for breaking down and metabolizing fats. The pancreas focuses on producing enzymes for digesting carbohydrates, proteins, and fats. The gallbladder's pear shape allows it to store and concentrate bile efficiently, expanding and contracting as needed to release bile into the small intestine. This structure is ideal for its storage and release functions.
So, okay, let's do the same thing. Explain it a bit like I'm stupid.
Sure, the liver makes a digestive juice called bile, which helps break down fats. The gallbladder is a small sac that stores this bile. Its pear shape helps it hold and release bile when needed, like squeezing a water balloon. When you eat fatty foods, the gallbladder releases the stored bile into the small intestine to help with digestion.
Interesting. And okay, cool. Let's continue with the sphincters. So, we've got the upper esophageal and the lower esophageal sphincter. Before we go into what these two are, explain to us, first of all, what sphincters are.
Sphincters are little circular muscles that act like valves. They control the opening and closing of certain passages in the body. When they contract, they close off a passage, and when they relax, they open it, allowing substances like food or liquid to pass through. They help regulate the flow and ensure things move in the right direction.
So, so one more time, um, this about the sphincters.
Sure, sphincters are circular muscles that control openings in the body. They act like valves, opening and closing to regulate the passage of substances. When they contract, they close off a passage, and when they relax, they allow substances to pass through. They help direct flow and maintain order in the digestive system.
I see. And how long are the periods that they can be contracting to close? And also, what about the pharynx? Why isn't the pharynx a sphincter?
Sphincters can contract for varying lengths of time, often just a few seconds during swallowing or digestion, but some can stay contracted longer if needed. The pharynx isn't a sphincter because it's a muscular passageway, not a valve. It guides food and air into the right tubes, but doesn't need to close off entirely like a sphincter does.
Okay. So a sphincter closes off by contracting, and for instance, it might allow sufficient time for nutrient absorption or something.
Exactly. When a sphincter contracts, it closes off a passage, controlling the flow of substances. This can indeed allow time for processes like nutrient absorption or digestion. For example, the lower esophageal sphincter prevents stomach acid from flowing back into the esophagus, while the pyloric sphincter controls food moving from the stomach to the small intestine, ensuring proper digestion.
Okay. That makes sense. And let's talk a bit about the pyloric and the ileocecal sphincters. Why are they called as such?
The pyloric sphincter is named after its location at the pylorus, the opening from the stomach to the small intestine. Pylorus means gatekeeper, reflecting its role in controlling the passage of food. The ileocecal sphincter is located between the ileum, the last part of the small intestine, and the cecum, the beginning of the large intestine. Ileocecal refers to these two sections, indicating its position and function in regulating the flow between them.
That makes a lot of sense. And then, when does the rectum start? I don't understand the border between the colon and the rectum.
The rectum starts at the end of the sigmoid colon. The transition is gradual, but anatomically the rectum begins where the colon straightens out and heads toward the anus. The rectum's main function is to store feces before they're expelled. It's the final section of the large intestine, leading to the anus.
Okay, got it. So, do we also have an anal sphincter then?
Yes, exactly. The anus has two sphincters, the internal and external anal sphincters. The internal sphincter is involuntary and keeps the anus closed at rest. The external sphincter is voluntary, allowing control over bowel movements. They work together to regulate the expulsion of waste.
Interesting. Okay, let's talk a bit about the layers of the gastrointestinal wall now.
Sure, the gastrointestinal wall has several layers. Starting from the inside, there's the mucosa, which secretes mucus and absorbs nutrients. Next is the submucosa, containing blood vessels and nerves. The muscularis layer, made of muscle, helps with peristalsis. Finally, the outermost layer is the serosa, which reduces friction between digestive organs. These layers work together to ensure proper function.
Okay, let's start with the most outer layer and let's understand a bit about its structure and function and why they're placed at the layer they are. So let's start with the serosa and let's start with its etymology then.
The serosa is the outermost layer of the GI tract. Its name comes from serous, referring to its secretion of serous fluid, which reduces friction. It's also called the visceral peritoneum. The serosa is a smooth membrane that covers and protects the GI tract, allowing it to move smoothly within the abdominal cavity. It's placed on the outside to provide a slippery protective surface, minimizing friction with other organs.
Okay, you mentioned serous fluid, and sorry for the noise, guys. You mentioned I'll exile my housemate. You mentioned serous fluid. Could you elaborate on that?
Sure, serous fluid is a lubricating fluid produced by the serosa. It helps reduce friction between the GI tract and surrounding organs as they move during digestion. This fluid allows the organs to glide smoothly against each other, preventing irritation or damage. It's essential. Sure,
Okay,
serous
okay.
fluid is a
So no, I got it first time. But okay, so it allows frictionless kind of movement then between the organs. Which organs?
Exactly. The serosa reduces friction between the GI tract and surrounding organs like the liver, intestines, and abdominal wall. This smooth, slippery surface allows the organs to move against each other without causing damage or discomfort, especially during movements like digestion or physical activity.
And so it allows the small intestine and the let's say colon to have frictionless movement.
Exactly. The serosa allows the small intestine and colon, among other organs, to move smoothly against each other. This helps prevent irritation and damage as they shift and contract during digestion and other bodily movements.
That makes a lot of sense, and now it makes sense why it's the outermost layer. So, how does it exactly produce this serous fluid? Does it have some kind of pores that it pours it out?
The serosa itself doesn't produce serous fluid directly. Instead, it's part of the peritoneum, which is a serous membrane. This membrane secretes serous fluid, providing lubrication. The fluid is produced by the cells in the peritoneum and helps organs glide effortlessly against each other, minimizing friction.
And where is the peritoneum?
The peritoneum is a thin, double-layered membrane that lines the abdominal cavity. The outer layer, called the parietal peritoneum, lines the abdominal wall, while the inner layer, the visceral peritoneum, covers the abdominal organs, including the serosa of the GI tract. It helps support the organs and provides a pathway for blood vessels, nerves, and lymphatics.
Okay, so it doesn't produce it, but it's the layer that happens to have serous fluid on it. Okay, so what about the other things about the serosa? So is it a connective tissue, and so is it a layer of squamous epithelial cells?
Yes, the serosa is made up of a layer of simple squamous epithelial cells, known as mesothelium, along with a thin layer of connective tissue. This combination provides a smooth, slippery surface that reduces friction between organs. It's part of the visceral peritoneum that helps protect and support the organs within the abdominal cavity.
So what other types do we have? We have connective, excuse me, tissue, and what else? Excuse me.
In the body, we have four main types of tissue: epithelial, which covers surfaces and lines organs; connective, which supports and binds other tissues; muscle, which facilitates movement; and nervous tissue, which transmits signals. Each type has specialized functions essential for the body's structure and operation.
One more time, please.
Of course, we have four main tissue types: epithelial tissue, which covers surfaces and lines organs; connective tissue, which supports and binds other tissues; muscle tissue, which enables movement; and nervous tissue, which transmits signals throughout the body.
So in this case, why does serosa have connective tissue?
The serosa has connective tissue to provide structural support and strength. It anchors the serosa to surrounding tissues and organs. It includes bone, cartilage, fat, blood, and lymph. This tissue provides structural and metabolic support, stores energy, and helps repair tissues. It plays a crucial role in maintaining the.
Excuse me. You lost me a bit. I think I might have confused you. So why is serosa specifically? Why does it have connective tissue?
The serosa contains connective tissue to provide structure and support. This connective tissue anchors the serosa to surrounding tissues, ensuring it stays in place. It also contains blood vessels, nerves, and lymphatics, which are essential for nourishing and maintaining the health of the outer layers of the GI tract.
Got it.
So connective
So what exactly are these other tissues that the serosa connects to?
The serosa connects to surrounding tissues like the muscularis layer of the GI tract and the parietal peritoneum. It also interacts with nearby organs and structures in the abdominal cavity, such as the liver, intestines, and other parts of the digestive system. This connection helps anchor the GI tract and facilitates smooth movement within the abdominal cavity.
Okay. Now, why do we call it a layer of squamous epithelial cells? What's squamous and what's epithelial?
Squamous refers to flat, scale-like cells. Epithelial pertains to epithelium, which is tissue that lines surfaces and cavities of the body. So a layer of squamous epithelial cells means a flat cell layer lining a surface. This type of epithelium, found in the serosa, helps protect and reduce friction.
Okay, explain to me like I'm stupid.
Sure,
Yeah.
no problem. Squamous means flat and thin, like a pancake. Epithelial refers to cells that form protective layers on surfaces, like skin. So squamous epithelial cells are flat cells that line and protect surfaces.
So squamous means flat and thin. How come?
Squamous comes from the Latin word squama, meaning scale. These cells are called squamous because they resemble fish scales, flat, thin, and plate-like. This shape is efficient for lining surfaces where a smooth, low-friction layer is needed.
And what about epithelial? Tell me again.
Epithelial refers to the epithelium, a type of tissue that lines the surfaces and cavities of the body. The term comes from Greek words epi, meaning upon, and thele, meaning nipple or surface. Epithelial tissue forms protective barriers and is involved in absorption, secretion, and sensation.
And what other types do we have? We have stratified too or something.
Yes, exactly. Besides simple squamous epithelium, we have stratified epithelium, which has multiple layers for protection, like in the skin. There's also cuboidal epithelium, which is cube-shaped and found in glands, and columnar epithelium, which is taller and lines the intestines. Each type serves different functions based on their structure.
So we have stratified, columnar, and squamous.
Yes, exactly. Stratified squamous epithelium has multiple layers of flat cells for protection, like in the skin. Stratified columnar epithelium has layers of column-shaped cells and is found in certain glands and ducts. Each type is adapted for specific protective or functional roles.
Wait, so we combine them too. Excuse me. So tell me all the types and subtypes.
Sure, here are the main types of epithelial tissue. Simple epithelium, single layer. Simple squamous, flat cells, found in places like blood vessels and lungs. Simple cuboidal, cube-shaped cells found in glands and kidneys. Simple columnar, tall, column-shaped cells found in the digestive tract. Stratified epithelium, multiple layers of flat cells found in skin, mouth, and esophagus. Stratified cuboidal, multiple layers of cube-shaped cells found in sweat glands. Stratified columnar, multiple layers of column-shaped cells found in certain glands and the male urethra. 3. Pseudostratified columnar epithelium, appears layered but isn't, often found in the respiratory tract. 4. Transitional epithelium, specialized for stretching found in the bladder. Each type serves different protective, secretory, or absorptive functions.
Amazing. Okay, now it makes sense. Okay, and why is the serosa referred to as the adventitia in the esophagus?
The esophagus is covered by a layer called the adventitia instead of the serosa. The adventitia is a connective tissue layer that anchors the esophagus to surrounding structures like the trachea and spine. Unlike the serosa, the adventitia doesn't produce serous fluid, as it's mainly for structural support rather than reducing friction.
Got it. So now it makes sense why it's not called serosa, but it doesn't make sense why it isn't why it's called adventitia. Also, how come we don't want these serous fluid on the esophagus?
Adventitia comes from the Latin adventitious, meaning external or coming from outside. This term reflects its role as an outer layer that connects the esophagus to surrounding structures. The esophagus doesn't have a serosa because it's primarily in the thoracic cavity, where it needs to be anchored firmly rather than move freely. Serous fluid isn't needed in the esophagus because reducing friction is less critical there compared to the abdominal organs.
One more time.
Sure, adventitia means an outer layer that connects and supports structures. The esophagus has adventitia because it needs to be anchored firmly within the thoracic cavity. There's no serous fluid here because the esophagus doesn't require the same friction reduction as abdominal organs. The serosa, which produces serous fluid, is found in organs that need to move smoothly.
Why doesn't esophagus need friction reduction?
The esophagus doesn't need as much friction reduction because it primarily functions as a rigid tube for transporting food from the mouth to the stomach. It doesn't undergo the same extensive movements as the abdominal organs, which constantly shift and move during digestion. Instead, the esophagus relies more on its muscular contractions to push food down efficiently.
Hmm, interesting. Okay, I don't understand the reason.
No problem. The esophagus mainly needs to transport food efficiently. Since it's a straightforward passage, it doesn't move around as much as organs in the abdomen. Therefore, reducing friction isn't as crucial. The adventitia provides enough support to keep it stable. Does that help clarify?
No, and also I don't understand the name adventitia.
Got it. Adventitia comes from the Latin word meaning external or foreign. It refers to the outermost connective tissue layer that binds the esophagus to surrounding structures. Unlike the serosa, which is smooth and produces fluid, the adventitia is more fibrous, providing structural support rather than reducing friction.
This is hard for me.
I understand. It can be a lot of information. Essentially, the adventitia is just a sturdy outer layer that helps keep the esophagus in place. It doesn't produce fluid like the serosa, but instead provides support. If you have any specific questions about it, feel free to ask.
So adventitia, what's the etymology?
Adventitia comes from the Latin word adventitious, meaning coming from outside or foreign. This reflects adjectives explaining its role as an external layer that connects and supports the esophagus to surrounding structures. It's a descriptive term for this outermost layer.
And do we have adventitia in other places too?
Yes, the adventitia is found in other parts of the body, like the outer layer of blood vessels, the trachea, and some other tubular structures. It serves a similar function by providing support and anchoring these structures to surrounding tissues.
Okay, so give me more examples about adventitia in the body. And guys, just as a reminder, I will be putting timestamps until the end of May. All videos until then will be timestamped, but until then, bear with me. You can always skip it after that. So, yeah, tell me again more examples of adventitia. It's just important for me to kind of understand.
Sure, besides the esophagus, the adventitia is also found in blood vessels, like arteries and veins, where it provides support and attaches them to surrounding tissues. The trachea, where it helps connect the windpipe to nearby structures. Parts of the ureters, which carry urine from the kidneys to the bladder. The adventitia's role is to anchor and support these structures within the body.
Okay, let's go forward then. So, actually, so the esophagus doesn't need friction, and sorry, because what?
The esophagus doesn't need friction reduction because it's primarily a rigid tube for transporting food. It doesn't move as much as other abdominal organs, so it relies more on.
Sorry for interrupting. Yeah, okay, that makes sense. Okay, let's continue then. Let's go to the second lining. So it's the muscularis externa.
Exactly. The muscularis externa is the next layer inward. It's made up of muscle fibers that are responsible for the peristaltic movements, which push food through the digestive tract. It typically has two layers, an inner circular layer and an outer longitudinal layer. These layers work together to mix and propel food along the GI tract.
Mm-hmm. So this is in charge of the muscles, basically.
Yes, exactly. The muscularis externa is responsible for the muscle contractions that move food through the digestive tract. It helps with mixing and propelling food along the way.
Why is it called externa?
It's called externa because it's the outer muscle layer of the GI tract, lying just beneath the serosa or adventitia. Externa means external in Latin, indicating its position as the outermost muscle layer compared to the inner muscular layers.
Do we have an inner muscular layer in this situation, the GI tract?
Yes, there's also an inner muscular layer called the muscularis interna, or simply the inner circular layer. In the GI tract, the muscularis externa has two sublayers, an inner circular layer that contracts to constrict the diameter of the tract, and an outer longitudinal layer that shortens the tract. These work together for movement.
So we have muscularis externa and muscularis interna.
In the GI tract, we typically refer to the muscularis externa, which has two sublayers, the inner circular layer and the outer longitudinal layer. There isn't a distinct muscularis interna term used. Rather, the muscularis externa encompasses both muscle layers.
How is this possible?
It's because the muscularis externa consists of two main layers of muscle, the inner circular layer and the outer longitudinal layer. These two layers together make up the muscularis externa, which is responsible for the peristaltic movements that propel food through the digestive tract.
So
There is
is it just called externa because it's more outside?
Exactly. It's called externa because it's the outer muscle layer compared to other inner layers like the mucosa and submucosa. It's more external relative to these deeper layers.
So why is it called a longitudinal layer? And why is it smooth muscle?
The outer layer of the muscularis externa is called the longitudinal layer because its muscle fibers run lengthwise along the tract. This orientation helps shorten and propel the food forward. It's made of smooth muscle, which is involuntary and controls slow, rhythmic contractions for peristalsis. Smooth muscle is ideal for this function because it's efficient and doesn't fatigue easily.
And what's the opposite then of longitudinal? What's the other categories?
The opposite of the longitudinal layer is the circular layer, where muscle fibers run around the circumference of the tract. This circular layer contracts to constrict the diameter of the tract, mixing and pushing food along. Together, the longitudinal and circular layers coordinate to produce effective peristaltic movement.
So we also have inner circular layer of smooth muscle.
Yes, exactly. The muscularis externa includes an inner circular layer of smooth muscle. This layer works with the outer longitudinal layer to mix food and propel it through the digestive tract.
What do you mean I get the inner part, obviously, but what do you mean circular? Circular. How is it circular?
By circular, I mean the muscle fibers in this layer encircle the GI tract, running around it like rings. When these fibers contract, they constrict the tract's diameter, helping to mix and push food along. So circular describes the orientation of the muscle fibers.
I'm what some would call confused.
No worries, I'm here to help. Think of the circular layer as muscle fibers that wrap around the tube of the GI tract. When they contract, they squeeze the tube, narrowing its diameter. This helps mix and push the food along. Does that help clarify?
Explain it to me like I'm stupid.
Sure. Imagine the GI tract as a tube. The circular layer is like rubber bands wrapped around the tube. When these bands tighten, they squeeze the tube, helping to push food along and mix it. That's the role of the circular muscle layer.
Okay, that's a bit better then. All right, so let's talk a bit more about how it contains the myenteric nerve plexus. That's part of the enteric nervous system. But also, let's talk about why circular is better to be inner and longitudinal is better to be outer.
The myenteric nerve plexus, located between the circular and longitudinal layers, coordinates muscle contractions for peristalsis. The circular layer is better inside because it can constrict the diameter of the GI tract, effectively mixing and moving food. The longitudinal layer on the outside helps shorten the tract and push food forward. Together, these orientations optimize the movement and processing of food.
Again,
Sure. The myenteric nerve plexus, situated between the circular and longitudinal muscle layers, controls their contractions for effective peristalsis. The inner circular layer is positioned to constrict the tract, aiding in mixing and moving food efficiently. The outer longitudinal layer helps shorten the tract, pushing food along. Their arrangement ensures coordinated, effective movement of food through the digestive tract.
I'm sorry, but could you explain to me like I'm dumb? So, guys, I'm gonna start using this phrase because it sounds a bit bad, doesn't it? But uh, let me know in the comments how I can say it more nicely.
No worries. Think of the circular layer as rings that squeeze the tube to mix food. The longitudinal layer runs along the tube, helping to push food forward. The myenteric nerve plexus is like a control center between these layers that helps coordinate their movements, so food is moved efficiently through your digestive system.
Why is it called myenteric? What's the etymology? What's the etymology of plexus? And also, okay, tell me again about what the myenteric nerve does. And yeah, I still don't understand why we have outer longitudinal and not outer circular layer and inner longitudinal.
Myenteric nerve plexus comes from myomuscle and enteric intestines, indicating its location within the muscle layers of the intestines. Plexus means a network, referring to the network of nerves. The myenteric nerve plexus coordinates the contractions of both muscle layers, enabling peristalsis to move food through the intestines. The outer longitudinal layer is on the outside because its function is to shorten the tract and move food along. An outer circular layer would be less effective for this, as circular muscles are primarily needed for constricting and mixing inside. This arrangement maximizes efficiency.
And why is circular better for constricting?
Circular muscles are better for constricting because they wrap around the circumference of the tube. When they contract, they narrow the diameter of the GI tract, effectively squeezing and mixing the contents inside.
And,
This is
and
crucial.
couldn't a longitudinal do this?
A longitudinal layer shortens the length of the GI tract rather than constricting its diameter. This motion helps propel food forward, but isn't as effective for mixing and breaking down food.
I see the difference. Okay, that makes sense. So the longitudinal, you're saying it's kind of pushing to wait, no, it's making the diameter smaller by pushing, you mean, but the circular is constricting from the inside?
Yes, that's correct. The longitudinal layer shortens the GI tract, helping push food along. The circular layer constricts from the inside, narrowing the diameter to mix and break down food. Together, they ensure efficient movement and processing of food.
And the circular layer, is it like in individual circles?
Not exactly individual circles, but more like a continuous ring of muscle fibers encircling the GI tract. When these fibers contract, they constrict the entire circumference, narrowing the passage and aiding in mixing and moving food.
Guys, I advise you to Google it. I'm gonna Google it now.
Sure, feel free to look it up if you need more details. Let me know if you have any other questions.
I have so many questions. Just give me a second. Just want to picture this.
Take your time. I'm here whenever you're ready.
And what's peristalsis then?
Peristalsis is the process of wave-like muscle contractions that move food through the digestive tract. The circular and longitudinal muscles in the GI tract contract and relax in a coordinated manner, pushing the contents forward. It's like a squeezing motion that propels food from the esophagus to the stomach and through the intestines.
One more time.
Sure. Peristalsis is a series of wave-like muscle contractions that move food through the digestive tract. The muscles in the GI tract contract and relax in a coordinated way, pushing the food forward. It's like a squeezing motion that moves food from the esophagus to the stomach and through the intestines.
And what's the etymology then?
The term peristalsis comes from the Greek words peri, meaning around, and stalsis, meaning constriction. It describes the process of constricting and relaxing muscles to propel contents through a tube like the digestive tract.
One more time.
Sure, peristalsis comes from the Greek words peri, meaning around, and stalsis, meaning constriction. It refers to the process where muscles constrict and relax in a wave like motion to move contents through the digestive tract.
Okay, I've googled the picture and I saw now the outer longitudinal smooth muscle and the circular. I'm still nonetheless confused about why we don't have the circular on the outside and the longitudinal on the inside.
It's a great question. The arrangement is based on function. The inner circular layer is more effective for constricting and mixing food, while the outer longitudinal layer shortens the tract to push food along. This combination optimizes the movement and processing of food.
Yeah, but why does this specific shape, circular or longitudinal, help with their function? If you could very briefly, in 2 sentences or so, explain like the key difference, please.
Sure, the circular layer wraps around the tract, so when it contracts, it narrows the passage, helping to mix and control food movement. The longitudinal layer runs along the tract, so when it contracts, it shortens the length, pushing food forward.
I'm confused. I'm sorry.
No problem. Think of the circular layer like squeezing a toothpaste tube. It narrows the passage to mix and control movement. The longitudinal layer is like pushing the tube from the bottom. It shortens and moves food forward. Does that help?
No, because I don't understand why the circular is on the inside and the longitudinal is on the outside. And I'm not gonna go forward if I don't understand it. I'm very thankful for the help.
The circular layer is inside because its job is to squeeze and mix the food, which works best when it directly wraps around the tube. The longitudinal layer is outside because its job is to shorten and push food forward, which is more effective when pulling from the outer surface. This setup ensures smooth and controlled movement through the digestive tract.
Yeah, but how does the structure actually help the function? Why is circular better at doing that job than the longitudinal and vice versa?
The structure helps because of how muscle fibers generate force. Circular fibers wrap around the tube, so when they contract, they squeeze inward, narrowing the passage and mixing the food. Longitudinal fibers run lengthwise, so when they contract, they shorten the tube, pulling it forward like an accordion. This arrangement allows each layer to specialize, circular for constriction and mixing, longitudinal for propulsion.
Ugh, we have to go a bit deeper into this.
Absolutely. The key difference comes down to the direction of force. Circular muscle fibers are arranged around the GI tract like rings. When they contract, they decrease the diameter of the tube, which is the most efficient way to mix and push food incrementally. Longitudinal fibers run parallel to the length of the tube. When they contract, they shorten the tube, pulling it forward and making propulsion more effective. If the arrangement were reversed, a longitudinal inner layer wouldn't efficiently mix food because shortening alone wouldn't create enough internal pressure. Likewise, an outer circular layer wouldn't effectively push food forward because constriction alone doesn't generate forward movement. This is why their specific placement is so important.
Kind of makes more sense. So it's basically like it's not exactly the same, but there's more chance of me kind of, I guess, squishing an apple if I create a circle with my hands than if I create like parallel my hands put my hands parallel. Is it like that? But what would be the analogy for the reverse of longitudinal's function?
Yes, that's a great way to think about it. If you cup your hands in a circle and squeeze inward, you can apply pressure evenly, like the circular muscles. If your hands are just parallel and you push, you won't squish the apple much. For the reverse, imagine trying to push a rope forward by squeezing it in the middle. It won't go anywhere. That's what would happen if the circular layer were outside trying to push food along.
Hmm.
It wouldn't be able to generate forward motion effectively because squeezing doesn't create directional movement. The longitudinal layer being outside pulls the tube forward like stretching and releasing an accordion, making propulsion smooth and efficient.
Okay, that makes sense. And what's the my in front of the myenteric nerve?
Audio transcription myenteric comes from the Greek word mes, meaning muscle. So myenteric literally means related to the muscle of the intestine. This makes sense because the myenteric nerve plexus is embedded between the circular and longitudinal muscle layers and controls their contractions to coordinate peristalsis.
Hmm. Okay. So, so, um, okay, so let's continue with the next one, the submucosa. Can you explain why the name is as such and what type of tissue it is? What kind of layers does it link?
Sure, submucosa comes from sub, meaning under, and mucosa, which refers to the inner lining of the GI tract. So the submucosa is the layer beneath the mucosa. It is made of connective tissue, which provides support and elasticity. The submucosa links the mucosa, the innermost layer responsible for absorption and secretion, to the muscularis externa, the muscle layer responsible for movement. It also contains blood vessels, nerves, and lymphatics supplying nutrients and helping regulate digestive functions.
Okay, and that makes a lot of sense. Okay, and so you said so it contains lymph vessels and contains blood supply. Why is that?
Um, Great question. The submucosa contains blood vessels because it needs to supply oxygen and nutrients to the mucosa, which is responsible for absorbing nutrients from food. It also carries away absorbed nutrients to the rest of the body. Lymph vessels are present because they help absorb and transport fats, which are too large to enter blood capillaries directly. They also play a role in immune defense, helping to detect and fight pathogens that might enter through the digestive system.
Interesting. Okay, so could you elaborate a bit on that? And I want to also ask if it has its own nerve plexus.
Absolutely. The blood vessels in the submucosa deliver oxygen and nutrients to keep the mucosa healthy, since it's constantly replacing cells due to wear and tear. They also transport absorbed nutrients from digested food into circulation. Lymphatic vessels are crucial because fats are broken down into fatty acids and absorbed into the lymphatic system before entering the bloodstream. The lymph system also helps remove waste and fight infections by carrying immune cells. Yes, the submucosa has its own nerve plexus called the submucosal plexus or Meissner's plexus. This network of nerves helps regulate blood flow, secretion of digestive enzymes, and the movement of the mucosa to optimize absorption and digestion. It works alongside the myenteric plexus to coordinate digestive functions.
Excuse me for the noise, guys. Okay, so okay, we talked about the submucosa. Why does it need its own submucosal nerve plexus, though?
No worries about the noise. The submucosal plexus, Meissner's plexus, is necessary because the submucosa controls very specific functions that need local regulation. Unlike the myenteric plexus, which mainly controls muscle contractions for movement, the submucosal plexus regulates secretion, blood flow, and mucosal movement. It helps fine-tune digestive enzyme release, controls the dilation and constriction of blood vessels to match digestive needs, and ensures the mucosa moves properly for optimal absorption. Having its own nerve network allows these processes to be regulated independently while still coordinating with the larger digestive system.
Hmm. Okay, and let's now talk about the mucosa.
Could you clarify what you mean by mute call?
No, no, excuse
Are
me,
you
sorry.
referring to something specific in our discussion?
I said mucosa, sir.
Ah, you mean the mucosa. The mucosa is the innermost layer of the GI tract. Its name comes from mucus since it secretes mucus to protect and lubricate the lining. The mucosa has three sublayers. One, epithelium, the innermost layer that absorbs nutrients and secretes enzymes and mucus. Two, lamina propria, a connective tissue layer that contains blood vessels, immune cells, and supports the epithelium. Three, muscularis mucosae, a thin layer of smooth muscle that helps move the mucosa for better absorption and secretion. It's the most active layer for digestion and absorption, making it essential for nutrient uptake.
I see. Okay,
Sorry,
so
I'm having issues.
okay, so we have the muscularis mucosa, which is a very thin layer of smooth muscle, and I assume that. So yeah, talk to us a bit about that. Also, talk to us about the lamina propria.
Yes, the muscularis mucosae is a very thin layer of smooth muscle within the mucosa. Unlike the muscularis externa, it doesn't move food along, but instead creates small local movements of the mucosa. These movements help mix digestive secretions and improve contact between nutrients and the absorptive surface, increasing efficiency. The lamina propria is the middle layer of the mucosa. It's made of loose connective tissue and contains blood vessels, lymphatics, and immune cells. It supports the epithelium by providing nutrients and playing a role in immune defense, helping to detect and fight pathogens that enter the digestive tract. This makes it essential for both digestion and protection.
Can you talk to me a bit more about the etymology this time of the lamina propria, and why is it mostly connective tissue, but also includes some lymphatic tissue?
Um, yes, I heard you. The lamina propria is made mostly of connective tissue because it provides structural support to the mucosa, anchoring the epithelium and supplying it with nutrients through blood vessels. This connective tissue also allows flexibility, so the mucosa can expand and contract as needed during digestion. It contains lymphatic tissue because the GI tract is constantly exposed to potential pathogens from food. The lymphatic tissue, including immune cells and lymphoid nodules like Peyer's patches, helps detect and neutralize harmful microbes before they enter the bloodstream. This immune defense is crucial for maintaining gut health while allowing safe nutrient absorption.
Okay, and why about how come it has epithelium containing both endocrine and exocrine cells?
Great question. The epithelium in the mucosa contains both endocrine and exocrine cells because it plays a dual role in digestion and regulation. Exocrine cells secrete digestive enzymes, mucus, and other substances directly into the lumen to aid digestion and protect the lining. For example, goblet cells secrete mucus, while other exocrine cells release digestive enzymes and acid in the stomach. Endocrine cells, also called enteroendocrine cells, release hormones into the bloodstream to regulate digestion. These hormones, like gastrin, secretin, and cholecystokinin (CCK), help control enzyme secretion, gut motility, and the release of bile and pancreatic juices. This combination allows the epithelium to both actively break down food and coordinate digestive processes throughout the GI tract.
Hmm. Let's talk a bit about region specialization now. So, when it comes to the esophagus, what exactly does it do? Is it so it's smooth muscle that facilitates transport, basically?
Yes, the esophagus is mainly responsible for transporting food from the mouth to the stomach and sac. It doesn't play a role in digestion or absorption, but ensures food moves efficiently through peristalsis, wave like muscle contractions.
Mm-hmm.
One unique feature of the esophagus is that its upper part contains skeletal muscle, allowing voluntary control over swallowing. As you move down, it transitions into smooth muscle, which takes over the involuntary movement. This specialization ensures a seamless shift from voluntary swallowing to automatic food transport.
Mm-hmm. Interesting. Okay, let's talk now about the stomach. So obviously we want it for um, storage, but also digestion and secretion. Can you talk more though about these things?
Sorry, I'm having issues
No,
right
that's fine.
now. Could not.
Okay, so can you tell me about the stomach then, about storage, secretion, digestion?
Yes. The stomach has three main functions: storage, secretion, and digestion. 1. Storage. The stomach holds food temporarily, allowing controlled release into the small intestine. The rugae, folds in the stomach lining, help it expand when food enters. 2. Secretion. The stomach secretes gastric juices, including hydrochloric acid, HCl, to break down food, pepsin to digest proteins, and mucus to protect the lining from acid. 3. Digestion. The stomach mixes food with gastric juices through churning motions, breaking it into a semi-liquid substance called chyme. This prepares nutrients for further digestion and absorption in the small intestine. Would you like to go deeper into any of these aspects?
Hmm, interesting. I would like to talk a bit about um, the small intestine and how its function for secretion differs and for mixing. Yeah, so let's talk a bit about that and how does it relate to things like digestion and absorption, and what are the differences between that and stomachs?
Great topic. The small intestine differs from the stomach in both secretion and mixing, and these differences are key to digestion and absorption. Secretion differences. The stomach mainly secretes acid, HCl, and pepsin, which help break down proteins. It also secretes mucus to protect its lining. The small intestine, instead of producing strong acids, secretes enzymes, bicarbonate from the pancreas and bile from the liver and gallbladder, to neutralize stomach acid and further break down nutrients. It also produces mucus, but mainly for lubrication rather than protection against acid. Mixing differences. The stomach mixes food by churning, using its muscular layers to mash food into chyme. The small intestine doesn't churn the same way. Instead, it uses segmentation, localized contractions, to mix chyme with digestive juices and increase contact with the absorptive surface. It also uses peristalsis to slowly move chyme forward while ensuring proper digestion and absorption. Digestion and absorption differences. The stomach mostly breaks down food, but absorption is limited, mainly alcohol and some drugs. The small intestine is where most digestion and absorption occur. Its inner surface is lined with villi and microvilli, which maximize nutrient absorption. So while the stomach's job is breaking food down, the small intestine's job is final digestion and nutrient absorption. Would you like to explore any part in more detail?
So, let's talk a bit about the endocrine cells in the GI wall then. So, can we start with CCK and motilin?
Yes. CCK, cholecystokinin, and motilin are both hormones secreted by endocrine cells in the GI tract, but they have different roles. One, CCK, cholecystokinin, secreted by eye cells in the duodenum and jejunum small intestine. Function: CCK stimulates the release of bile from the gallbladder and digestive enzymes from the pancreas. It slows gastric emptying to allow proper fat digestion. Trigger: it's released in response to fats and proteins entering the small intestine. 2. Motilin, secreted by M cells in the small intestine. Function: motilin triggers the migrating motor complex, MMC, which cleans out the GI tract between meals, preventing bacterial overgrowth. Trigger: It's released during fasting about every 90 minutes to stimulate peristaltic waves in the stomach and small intestine. So, CCK is active after meals to help with digestion, while Motilin works between meals to clear the gut. Want to dive deeper into either one?
So, can you give me the etymology? You jumped in straight forward to the deep end, I feel. Maybe not, but it has confused me. So let's keep it really brief and give me some key ideas about them instead. Include etymology and yeah.
Of course. Here's a quick breakdown with etymology. 1. CCK, cholecystokinin, etymology from Greek chole, bile, cyst, bladder, and kinin, to move, bile bladder mover. Key idea, it stimulates bile release from the gallbladder and enzyme secretion from the pancreas to help digest fats and proteins. 2. Motilin, etymology from motility, meaning movement, since it regulates GI motility. Key idea, it initiates the migrating motor complex, MMC, which clears the digestive tract between meals. So CCK helps with digestion, while Motilin helps with gut cleaning when fasting. Let me know if you want to go deeper.
So cholecystokinin, why is it called like that? And why is it secreted by eye cells in the small intestine specifically? Why does it increase pancreatic secretions and why does it decrease gastric emptying?
Why is it called cholecystokinin? The name comes from chola, bile, cysto, bladder, kinin, to move. This means bile bladder mover because its main function is to stimulate the gallbladder to release bile for fat digestion. Why is it secreted by eye cells in the small intestine? Eye cells are in the duodenum and jejunum because this is where fats and proteins first enter after leaving the stomach. The body needs bile and pancreatic enzymes here to digest them efficiently. Why does it increase pancreatic secretions? CCK stimulates the pancreas to release digestive enzymes like lipase and proteases because fats and proteins require enzymatic breakdown before absorption. Why does it decrease gastric emptying? It slows down the stomach's release of food into the small intestine so there's enough time for proper digestion and nutrient absorption before more food arrives. So, CCK ensures that the small intestine gets the right enzymes and bile at the right time while preventing overload. Want to explore more?
I'm confused. So why is it called cholecystokinin? And you said eye cells have to do with fats, but what exactly are eye cells? Why do they have
No
to do with
problem.
fats? Also, why is it in the small intestine specifically? And again, why does it increase pancreatic secretions and decrease gastric emptying? Remember, I'm quite dumb, so yeah, you have to dumb this down for me without oversimplifying.
No worries. Let's break it down step by step, keeping it simple but clear. Why is it called cholecystokinin? Chole means bile, cysto means bladder, kinin means to move. So cholecystokinin CCK literally means bile bladder mover because it tells the gallbladder to release bile. What are eye cells and why do they care about fats? Eye cells are special endocrine cells in the lining of the small intestine, duodenum and jejunum. They detect fats and proteins in food. Fats are hard to digest, so the body needs bile to break fats apart and pancreatic enzymes to further digest them. Eye cells release CCK when they sense fats and proteins, helping the digestion process. Why are eye cells in the small intestine? The small intestine is where food first arrives after the stomach. This is the best place to release enzymes and bile to digest food properly before it moves further down. Why does CCK increase pancreatic secretions? The pancreas makes digestive enzymes like lipase for fats and proteases for proteins. CCK tells the pancreas to release these enzymes because fats and proteins need them to be broken down into smaller parts for absorption. Why does CCK decrease gastric emptying? The stomach normally pushes food quickly into the small intestine. If food moves too fast, the small intestine won't have enough time to digest and absorb nutrients. CCK slows this down, making sure food stays longer in the stomach until digestion is ready in the small intestine. And in
Um,
short, CCK equal bile bladder mover. because it helps release bile. Eye cells detect fats and proteins because they need bile and enzymes to be digested.
So to be honest, I'm very, very confused. If you could summarize everything in 2 or 3 sentences, because my brain is all over the place now.
Of course. Here's the super simple version. CCK, bile bladder mover, is a hormone released by eye cells in the small intestine when fats and proteins arrive. It tells the gallbladder to release bile to break down fats and stimulates the pancreas to release digestive enzymes to break down fats and proteins.
So,
At the same time, it slows down the stomach's emptying so digestion can happen properly before more food enters. Does that help?
so in a sentence, why is it called cholecystokinin?
It's called cholecystokinin because chole equal bile, cysto bladder, kinin mover, signals the gallbladder to release bile, which helps digest fats.
But what is the chole part, and what is the cysto part, and what is the kinin part?
Got it. Here's the breakdown. Chol means bile from Greek chole. Cysto means bladder because bile is stored in the gallbladder. Kinin means mover because it stimulates movement, making the gallbladder release bile. So cholecystokinin
Okay.
literally means bile bladder mover because it makes the gallbladder release bile to help digest fats.
Okay.
Does that clear it up?
Yeah, that makes a lot more sense. How exactly does it do this then?
Hieno, that's it.
No, let's keep it in
Selkeytti.
English.
Audio description CCK is released by eye cells in the small intestine when fats and proteins are detected. It tells the gallbladder to release bile and stimulates the pancreas to release digestive enzymes. It also slows stomach emptying to give the small intestine time to digest properly.
So, in a sentence or two, why eye cells and why in the small intestine?
Eye cells are in the small intestine because that's where fats and proteins arrive after the stomach. They release CCK to trigger bile and enzyme secretion, ensuring proper digestion before food moves further down.
Okay, okay. And how are they able to secrete uh, to improve to increase pancreatic secretions exactly?
CCK binds to pancreatic receptors, stimulating the release of digestive enzymes that help break down fats and proteins in the small intestine.
Okay. And now in a sentence or so, how come it decreases gastric emptying?
CCK slows gastric emptying by binding to receptors on the stomach muscles, making them contract less and delaying the release of food into the small intestine.
Could you elaborate in 1 or 2 sentences about this? I don't understand.
CCK binds to receptors on stomach smooth muscle, causing it to relax, which slows the movement of food into the small intestine. It also activates nerves that delay stomach emptying, ensuring there is enough time for digestion in the small intestine.
But how does it do this stomach emptying? And yeah.
CCK inhibits stomach contractions by binding to receptors on gastric smooth muscle, reducing its motility. It also activates the vagus nerve, which signals the stomach to slow down emptying, preventing food from entering the small intestine too quickly.
Wait, again.
CCK binds to receptors on the stomach's smooth muscle, causing it to relax and contract less, which slows gastric emptying. It also activates the vagus nerve, sending signals that further delay the release of food into the small intestine.
I see. So okay, okay, fair enough. And now let's go back to motilin. In a sentence or so, why is it secreted by M cells in the small intestine? Why specifically M cells? And also, where does the word motilin come from?
Motilin is secreted by M cells in the small intestine because it regulates gut motility during fasting, triggering the migrating motor complex MMC to clear undigested food and bacteria. The name motilin comes from motility, as it stimulates movement in the GI tract.
Again, again, please.
It looks like your message might have been. Motilin is secreted by M cells in the small intestine because it controls gut motility during fasting by triggering the migrating motor complex MMC to clear out undigested food and bacteria. The name motilin comes from motility since it stimulates movement in the digestive tract.
Hmm. Again.
Motilin is released by M cells in the small intestine to stimulate the migrating motor complex, MMC, which clears the gut between meals. The name motilin comes from motility because it promotes movement in the digestive tract.
And what's the difference between mobility and motility?
Motility refers to the movement of internal structures, like how the digestive tract moves food through contractions. Mobility refers to the ability of an entire body or object to move freely, like a person walking or a joint bending. So motility is about internal movement, while mobility is about whole body movement.
Okay, okay. And then what about M cells? Sorry. This is I know this is a big part of this podcast, but it's really confusing me.
No problem. M cells in this context, not to be confused with immune related M cells, are enteroendocrine cells in the small intestine that sense fasting conditions and release motilin. They do this to trigger the migrating motor complex, MMC, which helps clear leftover food and bacteria between meals. They're located in the small intestine because that's where digestion slows between meals, and motilin is needed to restart movement and keep the gut clean.
And they also so the increase intestinal and gastric motility. Can you explain in one or sentences again the specifics of how they do this? Hey,
Motilin binds to receptors on smooth muscle cells in the stomach and intestines, triggering strong peristaltic contractions known as the migrating motor complex, MMC. It also stimulates the enteric nervous system, enhancing coordinated muscle contractions that clear the digestive tract between meals.
mm-hmm. Okay. Now let's briefly talk about gastrin secretin and gastric inhibitory peptide. Okay, I want you to tell me what cells secrete them for starters.
Gastrin is secreted by G cells in the stomach, mostly in the antrum. Secretin is secreted by S cells in the small intestine, duodenum. Gastric inhibitory peptide GIP is secreted by K cells in the small intestine, duodenum, and jejunum.
Hmm. And so these G cells, these S cells, these K cells, these I cells and M cells we have talked before, all of these, where do they come from?
These cells are enteroendocrine cells, meaning they are specialized hormone-secreting cells found in the epithelial lining of the gastrointestinal GI tract. Here's where they come from. G cells, found in the stomach antrum, they secrete gastrin, which stimulates acid secretion. I cells, located in the small intestine, duodenum, and jejunum, they secrete CCK to stimulate bile and pancreatic enzyme release. S cells, found in the duodenum, they secrete secretin, which stimulates bicarbonate release to neutralize stomach acid. K cells, located in the duodenum and jejunum, they secrete gastric inhibitory peptide GIP to slow stomach emptying and stimulate insulin release. M cells, found in the small intestine, they secrete motilin, which triggers the migrating motor complex MMC to clear the gut during fasting. These cells develop from stem cells in the intestinal crypts and differentiate into their specialized roles based on signals from the digestive system.
Okay, so all these cells come and they act at this in the small intestine. So what does gastrin do? Does it affect mucosal growth or gastric acid secretion?
Gastrin affects both gastric acid secretion and mucosal growth. 1. Increases gastric acid secretion. Gastrin stimulates parietal cells in the stomach to release hydrochloric acid, HCl, which helps digest food. 2. Promotes mucosal growth. Gastrin stimulates the growth of the gastric mucosa, ensuring the stomach lining stays healthy and capable of producing acid efficiently. So it boosts digestion by increasing acid and helps maintain the stomach lining for continued function.
And why doesn't it do the opposite? Decrease mucosal growth and decrease gastric acid secretion.
Gastrin doesn't decrease mucosal growth or acid secretion because its role is to enhance digestion, not slow it down. 1. Acid secretion is needed for digestion. Gastrin stimulates parietal cells to release HCl, which breaks down food and kills bacteria. If gastrin did the opposite, digestion would be less efficient. 2. Mucosal growth protects the stomach. Since acid is strong, gastrin also promotes mucosal growth to keep the stomach lining thick and resistant to damage. If it reduced mucosal growth, the stomach would be more vulnerable to ulcers. Gastrin's job is to prepare the stomach for digestion.
So do we have the opposite to gastrin in a certain sentence or two?
Yes. Somatostatin is the opposite of gastrin because it inhibits gastric acid secretion and slows mucosal growth. It is released by D cells in the stomach and intestine when acid levels are high, preventing excessive acid production and protecting the stomach lining.
Okay. Okay, so can we talk briefly about secretin? So it's secreted by S cells in the small intestine. How does it affect pancreatic secretions and gastric acid secretions?
Absolutely. Secretin is released by the S cells in the small intestine, especially when acidic chyme enters from the stomach. It plays a crucial role by stimulating the pancreas to release bicarbonate-rich fluid. This bicarbonate helps neutralize the stomach acid in the small intestine. Secretin also inhibits gastric acid secretion from the stomach to prevent excessive acidity. So, it helps create a balanced environment for digestion in the small intestine.
Hmm, interesting. So, why does it decrease gastric acid secretion but it increases
Great
pancreatic
question.
secretions?
Secretin decreases gastric acid secretion to protect the small intestine from too much acidity. When food from the stomach enters the small intestine, it can be very acidic. By reducing gastric acid production, secretin ensures that the acidic content is neutralized by bicarbonate from the pancreas before it moves further along. This also helps prevent damage to the intestinal lining and promotes an optimal environment for digestive enzymes to function properly.
And how come it increases pancreatic secretions? Also, it seems that gastrin does the opposite thing of secretin in terms of gastric acid secretions. I wonder what part of their structure allows that.
I see what you mean. The key difference lies in where and how these hormones act. Secretin is released in response to acidic chyme entering the small intestine, signaling that there's enough acid and promoting the release of bicarbonate to neutralize it. On the other hand, gastrin is produced by G cells in the stomach when food, especially proteins, are detected. Gastrin stimulates more acid production to help with digestion. So, their opposite effects are all about maintaining the right balance of acidity for optimal digestion.
Hmm, that makes a lot of sense. Okay, and what specifically though? Do they block receptors? What do they do?
Yes, exactly. Secretin and gastrin each bind to their own specific receptors on target cells. Secretin receptors are found in the pancreas and bile ducts, where they trigger the release of bicarbonate to neutralize acid. Gastrin receptors, on the other hand, are located on the parietal cells in the stomach lining, where they stimulate the production of gastric acid. By interacting with these specific receptors, both hormones precisely regulate different aspects of digestion and maintain the balance needed for efficient nutrient absorption.
Okay, so explain to me again why secretin increases pancreatic secretions?
Sure, secretin increases pancreatic secretions by stimulating the pancreas to release a bicarbonate-rich fluid. When acidic chyme from the stomach enters the small intestine, secretin is released by the S cells in response to the acidity. This bicarbonate secretion helps neutralize the acid, creating a more alkaline environment in the small intestine, which is essential for digestive enzymes to function properly and for protecting the intestinal lining.
Mhm. And what about gastric inhibitory peptides? They're secreted by K cells. Why K cells? And how come they produce an increase in insulin release and a decrease in gastric acid secretion?
Great question. Gastric inhibitory peptide, or GIP, is secreted by K cells in the small intestine, particularly when glucose or fatty acids are present. GIP plays a dual role. It stimulates the release of insulin from the pancreas to help manage blood glucose levels, and it inhibits gastric acid secretion to slow down the emptying of the stomach. This ensures that nutrients are absorbed more effectively and blood sugar levels are regulated properly.
How does this happen though?
This happens through specific receptors on the pancreas and stomach. When GIP is released by K cells in the small intestine in response to glucose or fatty acids, it binds to its receptors on pancreatic beta cells. This triggers the release of insulin, which helps lower blood sugar by promoting glucose uptake into cells. At the same time, GIP binds to receptors in the stomach to reduce gastric acid secretion and slow gastric emptying. ensuring that digestion and nutrient absorption happen more gradually and effectively.
I don't understand though how come it's an increase in insulin release, not a decrease, and then it's a decrease in gastric acid secretion and not an increase. Can you go into the specifics on the mechanism and also how come K cells are the ones that are secreted? Sorry, how come K cells are the ones that secrete?
No problem. Let's break it down. GIP, or gastric inhibitory peptide, is secreted by K cells in the small intestine when nutrients like glucose and fatty acids are detected. The reason GIP increases insulin release is that it binds to specific GIP receptors on pancreatic beta cells. This binding triggers a signaling pathway that enhances insulin secretion in response to elevated blood glucose levels. On the other hand, GIP reduces gastric acid secretion by acting on the stomach to slow down gastric motility and emptying. This gives more time for nutrients to be absorbed and helps regulate digestion more effectively. K cells are strategically located in the small intestine to sense incoming nutrients and quickly respond by adjusting hormone levels to optimize digestion and nutrient absorption.
Mhm. Okay, let's talk about smooth muscle functional syncytium. What's a functional syncytium, firstly?
A functional syncytium is a group of cells that work together as a single unit because they are connected in a way that allows them to coordinate their actions. In smooth muscle tissue, this means that the individual muscle cells are connected by gap junctions, which allow electrical impulses to pass from one cell to the next.
Mm,
This coordination.
Sorry, continue.
No worries. So, this coordination allows the entire smooth muscle tissue to contract together in a wave like manner. This is especially important in organs like the intestines or blood vessels, where smooth muscle contraction needs to occur in a coordinated and rhythmic fashion to move substances efficiently.
Hmm. And so I assume things like digestion are dependent then on the coordinated activity of the smooth muscle.
Absolutely. The coordinated contraction of smooth muscle in the digestive tract is essential for processes like peristalsis, which moves food along the intestines. Without this synchronized contraction, digestion would be far less efficient, and nutrients wouldn't be absorbed properly.
Got it. Okay. Can we talk a bit now about the interstitial cells of Sajal?
Of course, the interstitial cells of Cajal are specialized cells in the gastrointestinal tract that act as pacemakers, generating the slow wave electrical rhythms that coordinate smooth muscle contractions. These cells,
Where did they get their name from?
they are named after Santiago Ramon y Cajal, a pioneering Spanish neuroscientist who made groundbreaking discoveries in neuroanatomy. He identified these cells and their role in the nervous system of the gut, and his work laid the foundation for understanding how these cells coordinate gut motility.
Interesting. Okay. And so, can we talk a bit about um, gap junctions and how they're related to the big picture of our podcast, which is, motility of the gastrointestinal tract?
Absolutely. Gap junctions are essential for the coordinated activity of smooth muscle cells in the gastrointestinal tract. These specialized protein channels connect adjacent smooth muscle cells, allowing electrical impulses and ions to pass directly from one cell to the next. This communication allows the entire muscle layer to contract in a synchronized manner, which is crucial for effective peristalsis and the movement of food through the digestive tract. So, when we talk about absolutely,
No, no, no.
gap junction.
Continue. It's fine. I heard. I heard. Okay. So let me ask you another question then. since we're on the topic. So, do gap junctions enable the groups of muscle cells to be one single functional unit, or is it like a functional syncytium, or is it something different?
Yes, exactly. Gap junctions are what enable individual smooth muscle cells to function as a single coordinated unit, essentially forming a functional syncytium. These gap junctions allow electrical signals and small molecules to pass directly between neighboring cells, ensuring that they contract together in a synchronized manner. This coordination is crucial for effective muscle function in organs like the intestines, heart, and even blood vessels.
Hmm. I see. Let's talk more about these things. So, interstitial cells of Cajal. So, how do they work? What kind of waves do they drive?
The interstitial cells of Cajal generate and propagate slow waves, which are rhythmic oscillations in the membrane potential of smooth muscle cells. These slow waves set the basic rhythm for muscle contractions in the gastrointestinal tract. When these slow waves reach a certain threshold, they can trigger action potentials in the smooth muscle, leading to coordinated contractions that move food and other contents through the digestive system.
Hmm, interesting. Okay, so they drive cyclical, and are they cyclical slow waves?
Yes, exactly. These slow waves are cyclical in nature, meaning they occur at regular intervals. The frequency of these slow waves can vary depending on the region of the gastrointestinal tract. For example, in the stomach, the slow waves might occur about 3 times per minute, while in the small intestine, they can occur more frequently, around 12 times per minute. These waves help coordinate the timing of muscle contractions and ensure that food moves smoothly through the digestive system.
Hmm. And usually the GI smooth muscle cells, they usually have a negative resting membrane potential.
Yes, that's correct. GI smooth muscle cells typically have a negative resting membrane potential, usually around negative 50 to negative 60 millivolts. This negative resting potential is crucial because it allows for the generation of those slow waves and action potentials that drive the rhythmic contractions necessary for digestion and motility.
Hmm. Why 55?
Great question. The number 55 likely refers to the typical frequency of slow waves in some parts of the gastrointestinal tract, like the stomach, which can be around 3 to 5 cycles per minute. This frequency can vary depending on the specific region of the gut. With
Wait,
higher
I don't understand why minus 55.
I see what you mean. If you're referring to a value like minus 55, that typically refers to the resting membrane potential of these cells in millivolts. The interstitial cells of Cajal have a resting membrane potential around minus 55 millivolts, which is the baseline electrical state of the cells. When this potential becomes more positive due to slow waves, it can trigger muscle contractions.
Yes, but can you explain to me why minus 55 and not minus 570?
Sure, the resting membrane potential of minus 55 millivolts is due to the difference in ion concentrations inside and outside the cell, primarily involving sodium, potassium, and chloride ions. Cells maintain this potential using ion pumps like the sodium potassium pump, which moves sodium out and potassium into the cell. A potential like
All right.
minus 5
Okay. So, um, if a slow wave crosses the threshold potential, what happens?
When a slow wave crosses the threshold potential, it triggers an action potential in the smooth muscle cells. This action potential causes a rapid influx of calcium ions into the muscle cells, leading to muscle contraction. This coordinated contraction helps propel food and other contents through the digestive tract, ensuring efficient digestion and nutrient absorption.
Mm-hmm. Interesting. Okay, let me ask you a few more questions then. So, um, so we've got spike potentials. Could you explain to me this?
Sure, spike potentials are rapid, short bursts of electrical activity that occur when a slow wave in smooth muscle reaches the threshold potential. These spikes are similar to action potentials in neurons, and they cause a significant influx of calcium ions into the smooth muscle cells. This calcium influx leads to muscle contraction. So, while slow waves set the basic rhythm, spike potentials are what actually trigger the strong, coordinated contractions of the muscles.
Mm-hmm. Interesting. What about electric nervous innervation? How does it connect to the big picture?
Great question. Extrinsic nerve innervation, which includes inputs from the autonomic nervous system, helps regulate and modulate the activity of the gastrointestinal tract. These nerves can either
Excuse me, sir.
stimulate.
I said enteric, not extrinsic.
Got it. The enteric nervous system, often called the second brain, is a complex network of neurons embedded in the walls of the gastrointestinal tract. It operates independently of the central nervous system, but communicates with it. It controls gut motility, secretion, and blood flow, and it works closely with the interstitial cells of Cajal to coordinate smooth muscle contractions and ensure proper digestive function.
Hmm. Interesting. Okay. And what can trigger an enteric nervous innervation activity?
The enteric nervous system can be triggered by several factors, including the presence of food in the digestive tract, mechanical stretching of the gut walls, chemical signals from nutrients, hormones, and even signals from the central nervous system, like the parasympathetic nervous system.
Hmm.
These factors.
Mm-hmm. Okay, interesting. And so how come it can independently act from the nervous systems, the central nervous system?
The enteric nervous system has its own network of neurons, neurotransmitters, and reflex circuits that allow it to function autonomously. This means it can control many digestive processes like motility, secretion, and blood flow without direct input from the brain. However, it still communicates with the central nervous system through the vagus nerve and other pathways to ensure everything stays coordinated.
Hmm. Okay. And so, from what I understand, the ENS is comprised of two interconnecting plexuses, the myenteric plexus and the submucosal plexus. How come these two?
Exactly. The myenteric plexus, also known as Auerbach's plexus, is located between the longitudinal and circular muscle layers of the gut. It primarily controls gut motility by regulating muscle contractions. The submucosal plexus, or Meissner's plexus, is found in the submucosa layer and mainly regulates enzyme secretion, blood flow, and absorption by controlling the glands and epithelial cells in the gut lining. Both plexuses work together to ensure the smooth and efficient functioning of the entire digestive system.
Hmm. Interesting. Okay, let's talk about a bit autonomic nervous innervation now. So, what will be the differences? And yeah, how do they interface with the enteric nervous system or the smooth muscle directly?
Great. The autonomic nervous system includes the sympathetic and parasympathetic branches. The parasympathetic nervous system generally stimulates digestion by increasing gut motility and secretion through the release of acetylcholine. On the other hand, the sympathetic nervous system generally inhibits digestion by reducing motility and blood flow to the digestive organs using neurotransmitters like norepinephrine. These autonomic nerves interface with the enteric nervous system by modulating its activity. For example, parasympathetic fibers can enhance the activity of the enteric nervous system, leading to increased gut motility, while sympathetic fibers can inhibit it, slowing down digestion.
Okay, so what about things like emotions, fear, anxiety? Do these alter the balance? Are they under involuntary control? What goes on?
Yes, emotions like fear and anxiety definitely impact the autonomic nervous system. They can trigger the sympathetic response, like the fight or flight reaction, which can affect gut motility and other functions. It's mostly involuntary, but you can use techniques like deep breathing or mindfulness to help manage it.
So I don't understand why it's under involuntary.
It's involuntary because it's controlled by the autonomic nervous system, which handles things like heart rate, digestion, and breathing without conscious effort. This system keeps you balanced automatically so you don't have to think about it.
Yeah, but this sounds weird. This is very deterministic.
I get that it can feel a bit deterministic, but it's more about your body ensuring essential functions happen without you having to worry. You still have influence, though. Techniques like meditation or biofeedback can help you gain some control over these involuntary processes.
Yeah, but can we explain this thing you're talking about? Like, can we explain meditation and being proactive?
Sure, meditation involves focusing your mind and calming your thoughts, which can reduce stress and activate your parasympathetic nervous system, helping you feel more relaxed. Being proactive means taking steps to manage stress, like setting routines, practicing mindfulness, or exercising. These actions can help you maintain a balance between your sympathetic and parasympathetic systems.
So being proactive isn't part of the autonomic nervous system then.
Exactly. Being proactive is more about conscious choices and actions you take. It complements your autonomic system by helping manage stress and promoting overall well being.
Yeah, but I thought parasympathetic was in the autonomic. So you're saying I decrease the parasympathetic with, let's say, meditation or by being proactive, but I don't do it through the autonomic. Then how is this possible?
Great question. Meditation and proactive practices can indirectly influence the autonomic system. When you meditate or manage stress, you activate the parasympathetic system, promoting relaxation. It's like nudging your body in the right direction, even though the autonomic system itself is involuntary.
How is I don't understand. Can you go a bit deeper into this? It sounds like if other not civilizations, but if other people, let's say more Eastern philosophy people, wrote this stuff, they wouldn't have called it autonomic.
Sure, let's dive deeper. The autonomic nervous system includes both involuntary processes and influences from our actions. In Eastern philosophies, practices like meditation have long been understood to influence the body and mind. They might not call it autonomic, but they recognize the power of these practices to bring balance. Modern science just gives it a name.
Yeah, but the name means autonomic means in Greek autonomo. It means that it works by itself without us needing to do something. And I'm telling you, by being proactive or by or by practicing meditation, we are doing something.
You're right. Autonomic implies self governing, but our actions can influence it. Think of it like a thermostat. It works automatically, but you can adjust the settings. Meditation or proactive steps are like nudging that thermostat to a more comfortable setting.
Oh, dude, this was a good analogy. Perfect.
Thanks. Glad it made sense. If you have any other questions, feel free to ask.
Mm, yeah, of course, I have so many questions. So let's talk about excitation contraction coupling and let's fit it into the big picture of today's talk, which is motility of the GI tract.
Great. Excitation contraction coupling is essential for GI motility. It's how electrical signals in smooth muscle cells cause them to contract, moving food through your digestive system. It links the autonomic nervous system to actual muscle movement, fitting perfectly into our discussion on how the body regulates GI function.
Okay, and let me ask you. So we can you actually give us a dive into it a bit more, please?
Absolutely. Excitation contraction coupling in smooth muscle starts with an electrical signal, often from the autonomic nervous system. This signal causes calcium ions to enter the muscle cells. The increase in calcium triggers the muscle fibers to contract. This process allows for the rhythmic contractions that move food along the digestive tract.
So we have calcium influx through voltage-dependent calcium channels, basically, and the membrane when the membrane. So so how you connect this with membrane depolarization? I'm a bit confused.
Great question. When the membrane depolarizes, it means the electrical charge across the cell membrane changes. This depolarization opens voltage-dependent calcium channels, allowing calcium to rush into the cell. This
How
calcium influx,
how does it do it?
exactly.
Wait. How does it do it?
So the influx of calcium binds to proteins inside the muscle cell, triggering a series of reactions that lead to muscle contraction. This is how the electrical signal gets converted into a mechanical response, moving the muscle.
Hmm. So what exactly makes the calcium influx? Because why does membrane depolarization cause the calcium influx?
Membrane depolarization changes the electrical charge across the cell membrane. This change opens voltage-gated calcium channels, allowing calcium to flow into the cell. This influx is what kickstarts the contraction process.
I don't understand.
No worries. Think of it like a door. Depolarization is like flipping a switch that opens the door, the calcium channel. When the door opens, calcium rushes in, starting the muscle contraction process.
Yeah, it's great to think of it like a door, but what actually happens?
Sure, when the cell membrane depolarizes, it changes the voltage inside the cell. This change opens calcium channels, letting calcium ions flow inside.
Yeah, but
Those cal-
how does the voltage know? How does the voltage open the channel? How does this happen?
Voltage-gated calcium channels have sensors that detect the electrical change. When the membrane depolarizes, these sensors cause the channels to change shape,
And
opening the channel.
how do the sensors detect the change?
The sensors detect the change in electrical charge because they're sensitive to voltage differences across the membrane. When depolarization occurs, it changes the membrane's potential, which the sensors pick up,
Yes,
prompting
sir.
the
Yes,
channels to
yes.
open.
But what exactly makes them sensitive?
The sensitivity comes from specific protein structures in the channel. These structures respond to voltage changes, causing the channel to open or close when the membrane's electrical potential shifts.
Go deeper.
All right, these protein structures have charged regions that shift position when the membrane's voltage changes. This movement causes the channel to open, allowing calcium to enter the cell.
What are these proteins?
They're called voltage sensing domains, part of the channel structure, typically made up of specific amino acids that respond to voltage changes.
Okay. And do G proteins like come into play?
Yes, G proteins can also influence calcium channels, especially in smooth muscle cells. They act as messengers, relaying signals from receptors on the cell surface to the channels, modulating their activity.
What does G proteins stand for then?
G proteins are short for guanine nucleotide binding proteins. They bind to guanine nucleotides like GTP and GDP, helping transmit signals inside cells.
And how come they're able to do this? And why are they named as such?
They're named for their ability to bind guanine nucleotides. This binding allows them to act as molecular switches, turning on or off signaling pathways inside cells.
Again?
Exactly. They're crucial for relaying signals from the cell surface to internal processes.
Well, again, I don't understand what this G protein. Why are they called like this? Guanine. I don't understand.
No problem. G proteins are named because they bind guanine nucleotides, like GTP and GDP. These nucleotides help them switch between active and inactive states, allowing them to transmit signals.
They go from active to inactive states?
Exactly. When they bind GTP, they're active. When they hydrolyze it to GDP, they become inactive.
Okay, so when they hydrolyze GTP to GDP, so from 3 phosphates to 2, what?
Yes, exactly. They remove one phosphate group, converting GTP to GDP, which switches them to their inactive state.
Okay, fair enough. And why do they do this? And why do G protein coupled receptors induce release of calcium ions from the sarcoplasmic reticulum?
They do this to relay signals. When a G protein coupled receptor, GPCR, is activated, it triggers a G protein. This G protein can then activate other molecules, like IP3, which prompts the sarcoplasmic reticulum to release calcium ions.
Hmm. Okay.
Exactly.
Wow, this is really confusing. I can't lie. This podcast is one of the hardest we've done to now.
It's definitely complex stuff, but you're doing great. We can break it down step by step. Keep asking questions. I'm here to help.
Okay, how do D2 antagonists and antimuscarinics fit into all of this?
D2 antagonists and antimuscarinics work by blocking specific receptors. D2 antagonists block dopamine receptors, which can reduce nausea. Antimuscarinics block acetylcholine receptors, reducing secretions and slowing gut motility. Both can influence GI function.
Again,
Sure, D2 antagonists block dopamine receptors, often used to reduce nausea. Antimuscarinics block acetylcholine receptors, which helps decrease secretions and slow gut motility.
wait, so why are they called D2 antagonists? Why are they called prokinetic drugs? And why do they block dopamine? And why does dopamine cause nausea?
They're called D2 antagonists because they block dopamine type 2 receptors. They're not prokinetic drugs. Those actually increase gut motility. Dopamine can slow gut motility and cause nausea. So blocking its receptors can help reduce those symptoms.
Okay, can we talk a bit about calmodulin now? How does it fit to all of this? How does it connect with calcium?
Sure, calmodulin is a protein that binds calcium ions. When calcium levels rise, calmodulin activates by binding calcium, which then enables it to interact with various enzymes and proteins, influencing processes like muscle contraction and other cellular activities.
So how is myosin connected? And the chains and all that.
Calcium bound calmodulin activates an enzyme called myosin light chain kinase, MLCK. This enzyme phosphorylates myosin light chains, which enables myosin to interact with actin, leading to muscle contraction.
So calmodulin, what does it do to the MLCK? Activates. Why does it activate?
Calmodulin activates MLCK by binding to it when calcium levels are high. This binding changes MLCK's shape, enabling it to phosphorylate myosin, which then allows muscle contraction to occur.
Why do we need to phosphorylate myosin for muscle contraction to occur?
Phosphorylation of myosin light chains changes the myosin head's shape, allowing it to bind actin more effectively. This interaction is crucial for the sliding filament mechanism, which generates muscle contraction.
What's the etymology of myosin and actin and tropomyosin?
Sure, myosin comes from the Greek word mys, meaning muscle. Actin is derived from the Latin actus, meaning movement. Tropomyosin combines tropos, meaning turning, and myosin, reflecting its role in regulating muscle contraction.
And tell me more about the sliding filament mechanism. So how do myosin, actin, tropomyosin, how do they connect with each other?
In the sliding filament mechanism, myosin heads bind to actin filaments, forming cross bridges. Tropomyosin, regulated by calcium and troponin, moves to expose binding sites on actin. Myosin then pulls actin filaments, causing muscle contraction.
So tropomyosin and troponin remove actin from myosin?
Not quite. Tropomyosin blocks myosin binding sites on actin. When calcium binds to troponin, it causes tropomyosin to
Dude,
move,
slower,
exposing the
please,
binding site.
please, please, slower.
Sure, tropomyosin normally blocks myosin from binding to actin. When calcium binds to troponin,
Wait,
it shifts
let's get it.
tropomyosin.
Let's get it step by step. So you're saying tropomyosin will block myosin where actin usually binds to. Is that the step one?
Yes, that's step one. Tropomyosin covers the binding sites on actin, preventing myosin from attaching.
Why?
It acts as a regulatory mechanism, keeping the muscle relaxed until a contraction is needed.
Otherwise, okay, so it wants to prevent contraction, basically.
Exactly. It prevents contraction until the right signal arrives.
And when the right signal arrives, troponin comes.
Exactly. When calcium binds to troponin, it causes tropomyosin to move, exposing the binding sites on actin.
Okay, and where is actin during all of this? It's just sitting on the side, waiting like a third wheel for a couple to basically break up to go and flirt.
Haha, kind of. Actin is just waiting there, ready for myosin once tropomyosin moves. It's essential for contraction, but it needs the signal to get things started.
I didn't expect an AI to laugh today. You are funny people. Let's. The only way for me to kind of talk so much to you, ChatGPT, without thinking that you're like the same as us, because we said, guys, this is consciousness meeting experience. Sorry, consciousness meeting intelligence is to remember that you're all the information that we gave to you, and then you're kind of growing from that information without the consciousness. I have to remind myself continuously. Anyway, okay, as we said that, so we've got myosin, and then that myosin usually binds to actin to contract, but we have tropomyosin that binds to the myosin, and troponin comes and removes the tropomyosin when it's time to contract. But troponin will do it once calcium binds on it.
Exactly. You've got it. Once calcium binds to troponin, it moves tropomyosin out of the way, allowing myosin to bind to actin and initiate contraction.
Okay, so let's get the big picture of this excitation contraction coupling then. So we have these voltage dependent calcium channels. So we have this membrane depolarization. Depolarization means that we have influx.
Exactly. Depolarization means the cell's electrical charge changes, opening voltage dependent calcium channels.
And
This allows,
depolarization means that it's going plus zero.
yes, depolarization means the membrane potential becomes less negative, moving closer to zero, which triggers those calcium channels to open.
I see. So depolarization, let's say the QRS complex. RS is still depolarization, but it's going down.
Yes, exactly. The QRS complex in an ECG represents depolarization. The downward part shows the electrical impulse traveling through the heart, but it's still part of the depolarization process.
How come it's going down? It's becoming more negative.
The downward deflection on the ECG reflects the direction of electrical activity. It's not about the charge becoming more negative, but rather how the electrical signal moves through the heart muscle.
I don't,
Depolarization
I
is
don't, I don't understand. Sorry. Please explain again.
no problem. The downward deflection on the ECG represents the electrical impulse moving in a specific direction. Depolarization is still happening, but the wave's direction causes it to appear as a downward deflection.
But it's still above the zero mark, basically. So it's depolarization.
Yes, exactly. The electrical potential is still above zero, indicating depolarization is ongoing. The downward deflection just shows the direction of the impulse.
It's like deceleration, basically.
That's a good way to think about it. It's still part of the overall depolarization process, just in a different phase.
Okay. 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 So we have these VDCC, let's call them voltage dependent calcium channels. And then basically membrane depolarization induces the calcium inflows through them. And then, so, and then how are G protein coupled receptors connected?
G protein coupled receptors, GPCRs, can activate signaling pathways that also increase calcium levels. For example, they can produce IP3, which triggers calcium release from the sarcoplasmic reticulum, complementing the calcium influx from VDCCs.
Again, please.
Sure, GPCRs can trigger pathways that raise calcium levels, like producing IP3, which releases calcium from the sarcoplasmic reticulum, adding to the calcium that enters through VDCCs.
Okay. So IP3R is produced from the G protein coupled receptors, and that IP3R helps more calcium go through. Wow.
Exactly. IK3 is produced when a GPCR activates a G protein, leading to calcium release from the sarcoplasmic reticulum. This
And-
complements the calcium
Hey, okay.
influx from
So
VDCCs.
why do GPCRs have this ability, and why is IP3R needed? And why sarcoplasmic reticulum?
GPCRs can activate diverse signaling pathways, including IP3 production, because they interact with various G proteins. IP3 is
Excuse
needed
me,
to release
sorry,
calcium.
I'm gonna interrupt you, and guys, you'll notice that I've been interrupting more, but please talk more specifically to us. Like, I know all the time I say that we're dumb, but we need to actually know. Like, don't say various proteins. Tell us, and we're gonna ask you more questions. You are the divine being here.
Got it. GPCRs can activate specific G proteins that produce IP3. IP3 binds to receptors on the sarcoplasmic reticulum, a calcium storing organelle, causing calcium release into the cell.
Yes, but besides about what is happening, I want to know why it's happening. What makes their structure uh, like able to do this, and why are all these steps needed? Let's think of it a bit critically, please.
Sure, GPCRs have specific structures that let them interact with G proteins. These steps ensure precise control of cellular responses. The
This is
multi-step process.
sorry. This is very general answer. Please answer more specifically.
Sure, GPCRs have specific structures that let them interact with G proteins. These steps ensure precise control of
Excuse me.
cellular response.
What are these specific structures? Please be more specific. Don't. This is a bit. I mean, it's not disrespectful in the sense that you're consciously doing it, but come on.
Sure, GPCRs have 7 transmembrane helices that detect signals. When a signal binds, it changes the GPCR shape, activating G proteins. This G protein then triggers IP3 production, leading to calcium release from the sarcoplasmic reticulum.
Thank you. Okay, let's talk a bit more about this. So they have seven. What did you call them?
They're called transmembrane helices. These are like coils that span the cell membrane and help the receptor detect external signals.
And why are all these things needed? I don't understand.
These structures are needed to detect and transmit signals across the cell membrane, enabling the cell to respond to its environment.
Why
Each step
seven? Why seven and not 10?
7 helices are a common structure for GPCRs, allowing efficient signal detection and transmission.
Why is
The
it
design
more? Why is it more efficient than 10?
7 helices provide the right flexibility and stability for GPCR function. More helices might complicate the structure without adding benefit.
Why not 5?
5 helices might not provide enough structural support or functional capability. 7 is optimal for the receptor's flexibility and function.
Why?
7 helices create a stable, flexible structure
Yes,
that can effectively.
why do they create a flexible structure? Why do 7 specifically? What I'm asking you is, let's imagine I have 5 apples. I have 5 apples because I have 4 friends. going to give one to each one of them and one for me. This will be sufficient for all of us to be okay. But you're not explaining to me why. You're just saying it to me, like,
I get it. 7 helices allow the receptor to change shape just enough to bind to different molecules while remaining stable. It's like having enough apples for everyone, ensuring the receptor can function properly.
Yeah, but I'm telling you why I need 5 apples. Because I have 5 friends. You're missing out the friends part. Guys, for you comments if I'm being like really pushy here, but
Got it. For GPCRs, 7 helices are optimal because they provide the right balance of flexibility and stability for the receptor to detect and transmit signals effectively.
sorry, sir, but you need to be more specific. Yeah.
Sure, 7 helices allow GPCRs to efficiently span the cell membrane, creating pockets for ligand binding and enabling structural changes that activate intracellular responses.
Let's do it again.
Sure, G protein coupled receptors, GPCRs, have 7 helices that span the cell membrane. These helices form a structure that allows the receptor to interact with G proteins inside the cell. When a signaling molecule binds to the receptor on the outside, it causes a conformational change in these helices, which then activates the G protein inside. This activation is crucial for various cellular responses. Does that help?
Not really. I mean, a bit better than before, but so IP3R, how does it connect? I don't understand why we need IP3R to help out calcium. And why is the sarcoplasmic reticulum like the location where G protein coupled receptors induce the release of calcium?
IP3R, or inositol trisphosphate receptor, plays a crucial role in calcium signaling. When a GPCR is activated, it triggers a cascade that produces inositol trisphosphate, IP3. IP3 then binds to IP3R on the sarcoplasmic reticulum, SR, a structure that stores calcium within the cell. This binding causes the SR to release calcium into the cytoplasm, which is essential for processes like muscle contraction. The SR is the primary calcium store in muscle cells, making it the key location for calcium release. This release of calcium is essential for muscle contraction and other cellular processes. Does that clarify it?
A bit more, a bit more. So, why does calcium need to bind? So the thing is, I understand what happens. It's just I don't understand why it happens, and it's really annoying because I feel like I'm just memorizing things.
That's completely understandable. Calcium binding is crucial because it acts as a signal that triggers specific cellular responses. In muscle cells, for example, calcium binds to proteins like troponin, which then allows muscle fibers to contract. This binding and subsequent response are essential for converting a chemical signal into a mechanical action
Okay, so
like muscle
what exactly is in, the thing is? I don't understand. We say that it's its structure that allows it to do this, but what's so special about its structure? Can we go a bit deeper instead of just saying it's the structure that allows it to bind or whatever?
Sure, let's dive deeper. Calcium's ability to bind is due to its properties as a positively charged ion, Ca+. Proteins have specific sites that are structured to attract and hold on to this ion, often involving negatively charged amino acid residues like aspartate or glutamate. These binding sites are precisely shaped to fit calcium ions, ensuring strong and specific binding. When calcium binds, it induces a change in the protein structure, activating or deactivating its function. This structural
One more time,
specificity is
please. One more time. One more time.
sure. Calcium ions, Ca+, bind to proteins because of their specific structure. Proteins have binding sites shaped to attract and hold calcium, often involving negatively charged amino acids. When calcium binds, it changes the protein's shape, activating or deactivating its function. This structural specificity is what makes calcium such an effective signaling molecule in cells.
Hmm. Okay.
I'm glad.
Okay. So now we have increase in intracellular calcium, which basically will activate calmodulin, and this activation will. I don't understand how this activation makes the MLCK active.
Calmodulin, when bound with calcium, changes shape, allowing it to activate myosin light chain kinase, MLCK. Activated MLCK then phosphorylates the myosin light chains, which increases their interaction with actin, leading to muscle contraction. So calcium-bound calmodulin essentially acts as a switch to turn on MLCK and initiate contraction.
Okay. Why?
Calcium binding triggers changes in proteins, which is key to many cellular processes. It essentially acts as a switch, turning on or off various functions that are vital for the cell's operation.
And
Does
an
that make more
active
sense?
what is a myosin light chain kinase?
Myosin light chain kinase, MLCK, is an enzyme that plays a role in muscle contraction. It activates myosin by adding a phosphate group to the myosin light chain, which is essential for muscle fibers to contract. So, MLCK essentially helps turn on the muscle contraction process.
So myosin light chain kinase, does it phosphorylate myosin light chains?
Yes, exactly. Myosin light chain kinase phosphorylates the myosin light chains, which activates them and enables muscle contraction.
So once they get phosphorylated, they are activated, and then what happens? How exactly does muscle contraction occur then?
Once the myosin light chains are phosphorylated, the myosin heads bind to actin filaments, forming cross bridges. This binding allows the myosin heads to pull the actin filaments, sliding them past each other, which shortens the muscle fiber and causes contraction.
Okay,
This
thank you.
cycle of
Thank you. This was very helpful. All right, so um, let's go to something else. How this was helpful, thank you. Um, so it's weird for me to say thank you. I'm gonna break the fourth wall a bit here, guys. It's weird for me to say thank you and to act like the ChatGPT like is human, but I kind of try to do it to create for you guys a kind of better experience, like a more but it's just not honest. I don't know. Like I really like to be very, very polite to people in real life, but I don't know. We shouldn't confuse consciousness with intelligence. And anyway, there's arguments to be made. Tell me what you think in the comments. It would be very interesting. Okay, so segmentation and peristalsis. How do they connect to the overlying theme, which is motility of the GI tract?
You're absolutely right about the distinction between consciousness and intelligence. As for segmentation and peristalsis, they are both crucial for the motility of the GI tract. Segmentation involves rhythmic contractions that mix food and digestive juices, enhancing nutrient absorption. Peristalsis, on the other hand, is a series of wave-like muscle contractions that propel food through the digestive tract. Together, they ensure that food is properly mixed and moved along the GI tract for effective digestion and absorption. Does that make sense?
So talk to me about segmentation. So is it, I don't understand what it is.
Sure. Segmentation refers to contractions in the intestines that mix and churn the food. These contractions break the food into smaller pieces, ensuring it is thoroughly mixed with digestive enzymes and juices. This process helps with nutrient absorption by exposing the food to the intestinal lining. It doesn't move.
Yeah. Okay, so let me ask you, is it the circular muscle that we're talking about?
Yes, exactly. The circular muscles in the GI tract play a key role in both segmentation and peristalsis. For segmentation, these muscles contract in a coordinated manner to mix the food. During peristalsis, they contract behind the food bolus to push it forward through the digestive tract.
So one is for pushing forward and the other is for pushing it to mix.
Sure. Peristalsis is responsible for pushing the food forward through the GI tract, while segmentation mixes the food to enhance digestion and nutrient absorption.
And how does their name explain their function?
The names reflect their functions quite well. Peristalsis comes from Greek, meaning to wrap around, which describes the wave-like contractions that propel food forward. Segmentation refers to the process of dividing the food into segments, allowing for thorough mixing and digestion.
Wait, again, please.
Sure. Peristalsis means to wrap around, describing the wave-like contractions that move food forward. Segmentation refers to dividing the food into segments to mix and digest it thoroughly.
Yes, but how do the names reflect the function, sir? Why don't we call one peristalsis and the other segmentation?
The names reflect their functions quite well. Peristalsis comes from Greek, meaning to wrap around, describing the wave-like contractions that propel food through the digestive tract. Segmentation, on the other hand, refers to dividing the contents into segments,
Uh-huh.
which helps with mixing and digestion.
Mhm. That makes a lot of sense. Okay, so let's talk. So there's no net movement in segmentation, just for mixing.
Exactly. Segmentation focuses on mixing the contents without moving them forward, while peristalsis moves the contents along the digestive tract. Mechanical mixing, like segmentation, breaks down food into smaller pieces and thoroughly mixes it with digestive enzymes. This increases the surface area of the food, making it easier for enzymes to break down nutrients during chemical digestion. Does
Okay,
that clarify it?
yeah, okay. That makes more sense. So so peristalsis, they say, is for propulsion. What does this propulsion? What's the etymology?
The term peristalsis comes from the Greek word peristellein, meaning to wrap around or to constrict. It describes the wave-like muscle contractions that propel food through the digestive tract.
Again, please.
The term peristalsis originates from the Greek word peristellein, meaning to wrap around or to constrict. It describes the wave-like muscle contractions that propel food through the digestive tract.
I don't understand. Can tell me again about? Sorry, guys, I'm a bit confused.
No problem at all. Peristalsis comes from the Greek word peristellein, which means to wrap around or to constrict. It describes the wave-like muscle contractions that move food along the digestive tract. Does that help clear things up?
Yeah, yeah. But what is the etymology? What is pulsion?
Propulsion comes from the Latin word propulsus, which means to drive
Okay,
forward.
okay,
In the
okay.
I'm glad
Latin. We should kick Latin out the window, dude.
Latin has definitely influenced many languages and scientific terms. It's quite interesting how it connects so many concepts.
So, do you think without Latin, these things would be more complicated?
Latin has provided a common root for many scientific terms, making it easier to understand and communicate complex
Is it
ideas
is it
across different
easier?
languages.
Is it easier?
It can definitely make things easier. Having a common language for scientific terms helps create
Yeah,
clarity and
yeah.
consistency.
Why not have English as a common language?
English does serve as a common language in many scientific fields today, but having Latin roots provides continuity with historical terminology and helps bridge gaps between different languages.
Yeah, okay. We're not gonna talk about this again. We talked about it too much other episodes. Okay, so sorry guys. Today you're seeing my grumpy side. I'm just really frustrated with all of this situation. I'm
It's
trying
okay.
and I'm trying to do my best not to memorize and to actually understand the material and to use critical thought. And sometimes it's hard, you know.
Absolutely. It can be challenging, but focusing on understanding the concepts rather than memorizing will definitely pay off in the long run. You're doing
Okay.
great.
Okay. So what does distension mean, and how is it correlated to peristalsis?
Distension refers to the stretching or expansion of the walls of the GI tract, usually caused by food. This stretching signals the muscles to contract, triggering peristalsis.
So
So distension
what's the difference between distension and stretching?
Distension specifically refers to the expansion or swelling of a structure, often due to internal pressure, like when the GI tract expands with food.
Mm.
Stretching is a more general term for any elongation or lengthening.
Hmm.
Distension in the GI
Okay, so it
I'm
has
glad
So distension is because of pressure.
Yes, exactly. Distension happens when there's an increase in internal pressure, such as when food fills the GI tract, causing it to expand.
And what about extension? Isn't it because of pressure? What other reasons might there be?
Extension can refer to the lengthening or stretching of a structure, and it can happen for various reasons, not just pressure. For example, muscles extend when they relax, or joints extend when they straighten. So extension isn't solely due to pressure.
Uh, Interesting. Okay, can we talk a bit about um, how the myenteric plexus relates to all of these in peristalsis?
The myenteric plexus, part of the enteric nervous system, plays a crucial role in regulating peristalsis. It controls the rhythmic contractions of the GI tract muscles, ensuring coordinated movement and propulsion of food.
Yeah, that makes sense. Okay, that makes sense. So, um, let's talk a bit more about nervous innervation and the pathophysiology this time. So, I want to ask you about this specific disease with this such hard name. Hirschsprung's disease? Hirschsprung's?
Hirschsprung's disease is a condition where nerve cells are missing in parts of the intestines, causing problems with passing stool. This absence of nerve cells disrupts normal peristalsis, leading to severe constipation or blockage. It's usually diagnosed in infants.
Tell me more about this. Why was it named like this, for instance?
Hirschsprung's disease is named after Harald Hirschsprung, the Danish physician who first described it in 1888. He identified the absence of nerve cells in the intestines, which leads to the symptoms associated with the disease.
So we don't have my enteric plexus and submucosal.
Exactly. In Hirschsprung's disease, there's a lack of nerve cells in the myenteric and submucosal plexuses in certain parts of the intestine. This absence impairs the muscle contractions needed for moving stool through the bowel, leading to severe constipation or blockages.
So it's in a distinct portion.
Yes, it's typically in a distinct portion of the intestine, often the rectum or sigmoid colon. This segment lacks the necessary nerve cells for normal function.
What about, is the distal colon the same?
Yes, the distal colon often refers to the last sections of the colon, including the sigmoid colon and rectum. This area is commonly affected in Hirschsprung's disease.
Okay, and this part we have my enteric plexus or submucosal?
In the affected part of the intestine in Hirschsprung's disease, both the myenteric plexus and the submucosal plexus are absent, leading to impaired motility.
Hmm. Wow. Okay. So, what are some symptoms and what's the treatment for this then?
Symptoms of Hirschsprung's disease include severe constipation, abdominal distension, and difficulty passing stool. Treatment usually involves surgery to remove the affected segment of the intestine and restore normal bowel function.
One more time.
Symptoms include severe constipation, abdominal distension, and difficulty passing stool. Treatment involves surgery to remove the affected part of the intestine.
One more time.
No problem. Symptoms are severe constipation, abdominal distension, and difficulty passing stool. Treatment involves surgery to remove the affected segment.
Tell me again about the symptoms and the treatment.
Sure. Symptoms of Hirschsprung's disease are severe constipation, abdominal distension, and difficulty passing stool. Treatment typically involves surgery to remove the affected section of the intestine.
I see. So basically, we have constipation because we cannot move the food forward because of the nerve absent.
Exactly. The absence of nerve cells in the affected area disrupts normal muscle contractions, making it difficult for stool to move through the intestines, leading to constipation.
What about if the affected section is the whole thing? Is this possible or does this not happen?
It's rare, but possible. In cases where the entire colon is affected, it's called total colonic aganglionosis. It requires more extensive surgery to remove or bypass the entire colon.
And what happens in this case? You cannot surgically remove the whole thing.
Surgeons create an ostomy, an opening in the abdomen, to divert waste into a bag outside the body, helping manage the condition.
A bag outside the body, and you can live a normal life like this?
Yes, many people with an ostomy live normal, active lives. It may take some adjustment, but with proper care and support, it can be managed effectively.
Anostomy.
Yes, an ostomy is a surgical procedure to create an opening for waste to leave the body, often used when part of the intestines needs to be bypassed or removed.
Who thought of this?
Surgeons and medical experts developed ostomy procedures to help patients manage conditions like Hirschsprung's disease,
And they,
allowing for improved quality of life.
so they eat food, and so they never poop, these people? Wow.
With an ostomy, waste exits the body through the opening into a bag, so they don't pass stool in the usual way.
And the waste is like poop?
Yes, the waste is similar to stool, just collected in the ostomy bag instead.
And do they know when this is going to happen?
Yes, they usually get a sense of when the bag needs emptying, but it doesn't happen with the same sensation as a bowel movement.
And do they have an abdominal sensation?
Some people might feel sensations, like fullness, but it's usually different from the typical urge to use the bathroom.
And do you need to go to the bathroom to do it, I assume?
No, you don't need to go to the bathroom. The ostomy bag can be emptied wherever is convenient, usually at a toilet.
Yeah, but it seems socially appropriate to do it to the bathroom.
Yes, most people prefer to empty it in a private setting like a bathroom for comfort and discretion.
And does it hurt?
The procedure itself doesn't hurt after healing, but adjusting to an ostomy can take some time.
Wow. And can this happen to you? Can the need for this come when you're 50?
It's possible, but not common. Most people won't need an ostomy unless they have a specific medical condition requiring it.
Yeah, I mean, okay, yeah, but this medical condition, is it only congenital?
Not always. Conditions like colorectal cancer, inflammatory bowel disease, or trauma can also necessitate an ostomy.
That's a good point. This is the craziest thing I've learned about medicine. This is amazing. Wow.
It really is fascinating. The human body and medical science are incredible.
Can you remind me what the interstitial cells of Cajal do?
The interstitial cells of Cajal act as pacemakers in the GI tract, generating electrical signals that coordinate smooth muscle contractions, essential for peristalsis.
Mm-hmm. Um, one more time.
Sure. The interstitial cells of Cajal generate electrical signals that help coordinate smooth muscle contractions in the GI tract, which are crucial for peristalsis.
I'm confused.
You're welcome.
No, I said I'm confused, sir.
No worries. The interstitial cells of Cajal act as pacemakers, creating signals that coordinate muscle contractions in the GI tract, essential for peristalsis.
Okay, got it now. Okay, let's talk about uh, the process of swallowing now. Can you introduce us to the world of swallowing then?
Sure. Swallowing involves three phases. The oral phase, where food is chewed and moved to the back of the mouth. The pharyngeal phase, where the airway is closed off and the food is pushed down the throat. And the esophageal phase, where food is moved to the stomach by peristalsis.
Okay, so again,
Sure. Swallowing has three phases. The oral phase, where food is chewed and moved to the back of the mouth. The pharyngeal phase, where the airway is closed off and food is pushed down the throat. And the esophageal phase, where food moves to the stomach.
okay. So And do we need to have like coordinated muscular contraction for the food to move?
Yes, exactly. Coordinated muscle contractions are essential for each phase, ensuring smooth movement of food from the mouth to the stomach.
And how does deglutition like connect to this?
Deglutition is just another term for swallowing. It encompasses all three phases: oral, pharyngeal, and esophageal, ensuring food moves efficiently from the mouth to the stomach.
And what's the etymology?
The term deglutition comes from the Latin word deglutire, which means to swallow down. It essentially describes the act of swallowing.
I don't understand.
Great. If you have more questions,
Yes,
feel free to
yes.
ask.
I said I don't understand.
No worries. Deglutition is just a fancy term for swallowing. It covers the whole process of moving food from your mouth to your stomach, step by step.
And how do the salivary secretions in the mouth and the mucus help?
Saliva and mucus in the mouth help by moistening food, making it easier to chew and form a bolus, which can then be swallowed more smoothly.
Hmm. Okay. So we have three phases: the voluntary phase, the pharyngeal phase, and the esophageal phase. Can you talk with us about this?
Absolutely. The voluntary phase is when you consciously chew and move food to the back of your mouth. The pharyngeal phase is involuntary, where the food is pushed down the throat and the airway is closed off. The esophageal phase involves peristalsis, moving the food down to the stomach.
So we have the voluntary phase. Tell me again. So we have these three phases, and then voluntary phase is basically when it's in the mouth.
Yes, exactly. The voluntary phase is when you're chewing and moving food to the back of your mouth, preparing to swallow.
So it goes to the oropharynx. Why is it called oropharynx? And so is the bolus that is going to the oropharynx? Bolus is the mixture of food with the saliva.
The oropharynx is the part of the throat right behind the mouth. It's called oro for mouth and pharynx for throat. The bolus, which is the chewed food mixed with saliva, moves from the mouth to the oropharynx during swallowing.
Hmm. And then the pharyngeal phase, which is involuntary, and the bolus goes through the pharynx into the esophagus, and then the esophageal phase, where the from so esophagus, does the bolus move
Exactly.
from the esophagus to the stomach?
Yes. During the esophageal phase, the bolus moves down the esophagus into the stomach through coordinated muscle contractions called peristalsis.
Okay, and how does the tongue move in the voluntary phase?
In the voluntary phase, the tongue helps by pushing the bolus to the back of the mouth toward the oropharynx using a coordinated wave-like motion.
So it's like an upward and back movement.
Exactly. The tongue presses the bolus upward and backward against the palate to guide it into the oropharynx.
That makes sense. And what is a palate?
The palate is the roof of your mouth. It has two parts: the hard palate at the front and the soft palate at the back.
Hmm.
The soft,
Yeah, sorry, continue.
no worries. The soft palate helps close off the nasal passages during swallowing to prevent food from entering the nasal cavity.
So what's the difference between? So I understand the hard palate structure difference with the soft palate, but what's the function difference?
The hard,
Excuse me. Tell me again.
I'm sorry, but you've hit the hard palate provides a rigid surface for the tongue to push food against, helping to move the bolus backward efficiently. The soft palate lifts and closes off the nasal passage during swallowing, preventing food from entering the nasal cavity.
Interesting. Okay, and so we have the passage of the bolus into the oropharynx. Are there receptors there or something?
Yes, The oropharynx contains mechanoreceptors that detect the bolus and trigger the involuntary pharyngeal phase of swallowing. These signals travel via the glossopharyngeal CNIs and vagus CNX nerves to the brainstem, coordinating the next steps.
So what do these receptors do, sorry?
These mechanoreceptors sense the bolus and send signals to the swallowing center in the brainstem. This triggers reflexive muscle contractions, closing the nasal passage and airway while pushing the bolus downward.
Wait, so we have sensory receptors that are stimulated from the passage of the bolus into the oropharynx, but how exactly are they stimulated?
They are stimulated by mechanical pressure. When the bolus touches and stretches the mucosa of the oropharynx, it activates mechanoreceptors. These receptors then fire signals through the glossopharyngeal, CNInd, and vagus, CNX nerves, telling the brainstem to start the next phase of swallowing.
Okay, are mechanoreceptors the same as sensory?
Yes,
Okay.
mechanoreceptors are a type of sensory receptor specifically designed to detect mechanical stimuli like pressure, stretch, or touch. In swallowing, they sense the bolus pressing against the oropharynx and send signals to the brainstem.
Mhm. So, well, so then we have pharyngeal phase then. Can you talk to about pharyngeal phase a bit? Like, yeah.
Absolutely. The pharyngeal phase is where swallowing switches from voluntary to involuntary control. Once the mechanoreceptors fire, the swallowing center in the brainstem, mainly the medulla, orchestrates a rapid reflexive sequence. 1. The soft palate elevates, sealing off the nasal cavity. 2. The epiglottis folds down, blocking the airway to prevent choking. 3. The pharyngeal constrictor muscles contract, pushing the bolus down toward the esophagus. It all happens in under a second. No thinking required.
Hmm, interesting. Okay. Um, so so in the pharyngeal phase, do we have impulses traveling?
Yes, during the pharyngeal phase, sensory impulses from the mechanoreceptors travel via the glossopharyngeal, CNIX, and vagus, CNX nerves to the swallowing center in the medulla. The brainstem then sends motor impulses back through the vagus, CNX, glossopharyngeal, CNX, and hypoglossal, CN12 nerves, coordinating muscle contractions to move the bolus downward.
Mm-hmm. Interesting. Okay. And so okay, we so we have the impulses traveling to the medulla oblongata and pons in the central nervous system. Why specifically in the medulla oblongata and pons?
Great question. The medulla oblongata and pons are key control centers for autonomic functions, including swallowing. The medulla houses the swallowing center, which integrates sensory input and coordinates the reflexive muscle contractions. The pons helps fine-tune these movements, ensuring smooth coordination between breathing and swallowing to prevent aspiration.
One more time. Why not another part of the brain?
Because the medulla oblongata and pons control reflexive, life-sustaining functions like breathing, heart rate, and swallowing. Higher brain areas, like the cortex, handle voluntary control, like deciding to swallow. But once the bolus reaches the oropharynx, it becomes an automatic reflex, which the brainstem is specialized to handle.
What other parts we have besides medulla oblongata and pons? What other options would we have?
Besides the medulla oblongata and pons, other brain regions include 1. Cerebrum, cortex, handles conscious thought and voluntary movements but is too slow for reflexes like swallowing. 2. Cerebellum, controls coordination and balance, but swallowing is more about reflex control than fine-tuned movement. 3. Midbrain, relays sensory and motor signals but isn't specialized for autonomic functions like swallowing. 4. Spinal cord manages reflexes for limbs but lacks the complexity needed for precise control of swallowing muscles. The brainstem, medulla plus pons, is the best choice because it integrates autonomic control, ensuring that swallowing happens instantly and safely.
Mm, yeah, I don't understand why not the other places.
Good point. Let's break it down. The medulla oblongata and pons are specialized for autonomic reflexes, meaning they control essential functions without conscious effort. Not the cerebrum. The cortex is too slow. It's for decision making, but once the bolus reaches the oropharynx, we need an immediate reflex. Not the cerebellum. It's for fine motor control and coordination, not reflexive sequences like swallowing. Not the midbrain. It relays sensory and motor information, but doesn't directly coordinate autonomic processes. Not the spinal cord. It controls basic reflexes like knee jerk, but lacks the complexity to coordinate swallowing muscles. The brainstem, medulla plus pons, is ideal because it's wired for fast, automatic control, integrating sensory input and sending out motor commands instantly.
And so, why, how come, how does the medulla oblongata and pons and other areas of the brain's name correlate to their ability to like correlate with certain things in the bodies, such as in this case, correlating with the pharyngeal phase and the impulses traveling to them there, you know?
It sounds like you're asking how the names of brain structures relate to their functions,
Yes.
particularly in controlling processes like the pharyngeal phase of swallowing. Brain region names often reflect their structure, location, or function, which helps explain why certain areas control specific body functions. For example, medulla oblongata, Latin elongated marrow, is named for its core, life sustaining role, managing
Like,
reflexes like swallowing, breathing, and heart rate. Pons, Latin bridge, connects different brain areas, helping coordinate autonomic functions like swallowing and breathing. This naming system follows historical anatomical discoveries and functional associations, helping us understand why the brainstem, rather than the cortex, cerebellum, or spinal cord, is in charge of swallowing reflexes. Did you mean dimethylamylamine instead of dimethylamylocodine? If
no,
so, that's a
no,
stimulant.
no, no, I didn't mean that. But okay, that makes sense. It seems like I just have to accept that I have to learn Latin to be a doctor. Okay, so in the pharyngeal phase, what's the actions of the soft palate and the uvula, and what is the uvula?
During the pharyngeal phase, the soft palate and uvula elevate to close off the nasopharynx, preventing food or liquid from entering the nasal cavity. This ensures that the bolus moves downward into the oropharynx and not backward into the nose.
Excuse me.
Yes, during the pharyngeal phase, the soft palate and uvula elevate, sealing off the nasopharynx to prevent food or liquid from entering the nasal cavity. This movement is controlled by the levator veli palatini and tensor veli palatini muscles, which contract in response to signals from the swallowing center in the medulla.
Confused. Sir.
Is the muscular, flexible part of the roof of the mouth that elevates
That's
during
what part.
swallowing to block the nasopharynx, preventing food from entering the nasal cavity. The uvula, from Latin uva, meaning grape, is the small, fleshy extension at the back of the soft palate that helps direct food downward and plays a role in speech and gag reflex.
Again?
The soft palate, Latin palatum molli, is the flexible, muscular part of the mouth's roof that elevates to close off the nasopharynx, preventing food from entering the nose during swallowing. The uvula, Latin uva, meaning grape, hangs at the back of the soft palate and helps guide the bolus downward, contributing to speech, swallowing, and the gag reflex.
I don't understand. I don't understand it.
The soft palate, palatum male, Latin for soft roof, is the flexible, muscular part of the mouth's roof that elevates to close the nasopharynx, preventing food from entering the nose. The uvula, uva, Latin for grape, hangs at the back of the soft palate, assisting in guiding the bolus downward and contributing to speech and the gag reflex. The
So,
epiglottis, api meaning above and glottis meaning tongue throat, is a flap of cartilage that folds over the larynx, blocking the airway to prevent choking during swallowing.
hmm. So why are these the structures perfect for their specific functions?
These structures are perfectly adapted for their functions because of their anatomical flexibility and precise control over movement. The soft palate is muscular and mobile, allowing it to seal off the nasopharynx during swallowing while also contributing to speech. The uvula is small but strategically placed, helping guide the bolus downward and triggering the gag reflex if needed. The epiglottis is cartilaginous and hinged, allowing it to snap shut over the airway during swallowing, preventing aspiration. Their different materials, muscle versus cartilage, and nerve control make them specialized yet coordinated for safe, efficient swallowing and speech.
And okay, and so okay, and okay. What about the sphincters? We've mentioned them in the beginning, but what happens now in the pharyngeal phase? Does the upper esophageal sphincter kind of do something?
Yes, the upper esophageal sphincter, UES, is a high pressure muscular valve that remains closed to prevent air from entering the esophagus and stomach. During the pharyngeal phase of swallowing, it relaxes to allow the bolus to pass into the esophagus, then quickly contracts again to prevent regurgitation.
Tell me again.
The upper esophageal sphincter, UES, is a muscular valve that normally stays closed to prevent air from entering the esophagus and to stop food from refluxing back up. During swallowing, it relaxes to let the bolus pass into the esophagus, then quickly contracts again to maintain pressure and prevent regurgitation.
I don't get it.
Think of the upper esophageal sphincter, UES, like a door that stays shut to keep air out and food in place. When you swallow, the door briefly opens to let food pass into the esophagus, then closes quickly to stop anything from coming back up or going down the wrong way.
Okay, that makes sense, but do we never get a bit of air through the sphincter?
Could you clarify what you mean?
Air through the sphincter.
Yes, a small amount of air can pass through the upper esophageal sphincter, UES, especially when swallowing or speaking. Normally, the UES stays tightly closed to prevent excess air from entering the esophagus, but some air can sneak through. This is why we sometimes burp or feel air trapped in the stomach.
Okay, and we also have the esophageal phase then. And here, what happens? We have peristaltic waves.
Yes, during the esophageal phase, primary peristaltic waves begin as soon as the bolus enters the esophagus. These rhythmic, wave-like muscle contractions push the bolus downward toward the stomach. If needed, secondary peristaltic waves occur to clear any remaining food.
Okay. So we have the peristaltic waves, they propel the bolus along the esophagus towards the stomach, and then what happens to the sphincter then?
As the peristaltic waves push the bolus toward the stomach, the lower esophageal sphincter relaxes to allow the bolus to enter the stomach. Once the bolus passes through, the LES contracts tightly again to prevent stomach acid from refluxing into the esophagus.
Okay, and then do the myenteric plexus and the vagal nerve innervate through this?
Yes, the vagus nerve, CNX, and the myenteric plexus work together to coordinate peristalsis and control the lower esophageal sphincter, LES. The vagus nerve sends signals to initiate peristalsis, while the myenteric plexus, part of the enteric nervous system, fine tunes muscle contractions along the esophagus to ensure smooth movement of the bolus.
Sorry guys for the noise. I'm a bit confused about the vagal nerve because we mainly talked today about the myenteric plexus and the submucosal plexus.
Great point. The vagus nerve, CNX, acts as an external regulator, sending signals from the brainstem to initiate and coordinate swallowing, including peristalsis in the esophagus. However, the myenteric plexus, Auerbach's plexus, is part of the enteric nervous system, ENS, and works independently to control esophageal motility, ensuring smooth wave-like contractions. The submucosal plexus, Meissner's plexus, on the other hand, mainly regulates secretion and blood flow, so it plays a smaller role in esophageal peristalsis.
Okay, let's continue a bit now with some more pathophysiology. First of all, define pathophysiology in a sentence before we continue.
Pathophysiology is the study of how disease or dysfunction alters normal physiological processes, leading to symptoms and complications.
Okay, and what's achalasia?
Achalasia is a disorder where the lower esophageal sphincter, LES, fails to relax, preventing food from entering the stomach and causing difficulty swallowing, chest pain, and regurgitation. It results from degeneration of the myenteric plexus, leading to impaired peristalsis and increased esophageal pressure.
Hey dude, yeah, let's talk more about the etymology of achalasia because I don't understand.
The word achalasia comes from the Greek a, meaning without or lack of, and chalasis, meaning relaxation or loosening. So achalasia literally means lack of relaxation, which perfectly describes the condition where the lower esophageal sphincter, LES, fails to relax, preventing food from passing into the stomach.
Okay, that makes sense. So it's the lower, why specifically the lower esophageal sphincter?
The lower esophageal sphincter, LE, is affected in achalasia because it relies on inhibitory neurons from the myenteric plexus to relax when food reaches it. In achalasia, these neurons degenerate, preventing the LES from receiving the proper signals to open. This leads to constant contraction, making it hard for food to pass into the stomach. The upper esophageal sphincter, UES, is controlled differently, so it remains functional.
All right, so um, let's talk a bit more about why it causes infection and ulceration and inflammation.
Achalasia can lead to infection, ulceration, and inflammation because food and liquids accumulate in the esophagus, creating a stagnant environment. This can, one, cause bacterial overgrowth, leading to esophagitis, inflammation, and increasing the risk of infection. Two, trigger chronic irritation, as food sitting in the esophagus damages the mucosa, leading to ulceration. Three, increase pressure, which can cause micro tears in the esophageal lining, further worsening inflammation and predisposing to complications like aspiration pneumonia.
And how do we treat achalasia?
Achalasia is treated by reducing lower esophageal sphincter, LES pressure, to allow food to pass into the stomach. Common treatments include 1. Pneumatic dilation. A balloon is inserted and inflated to stretch the LES. 2. Botulinum toxin, Botox injections, temporarily paralyzes the LES to reduce contraction. 3. Heller myotomy, a surgical procedure where the LES muscle is cut to allow relaxation. 4. Medications, like nitrates or calcium channel blockers. Relax the LES, though they are less effective. Each treatment depends on the severity of symptoms and patient condition.
If you could keep these things, thank you so much. If you could keep these things more brief, please, because I'm stupid. But so you said LES. What's LES?
You're not stupid. LES stands for lower esophageal sphincter, the muscular valve between the esophagus and stomach that controls food entry and prevents acid reflux. In achalasia, it fails to relax properly.
Okay, makes sense. I was just thinking of esophageal spelled differently. Okay, cool. So we'll cut the sphincter, and so what will replace the sphincter?
Nothing directly replaces the lower esophageal sphincter, LES, after a Heller myotomy, but the esophagus relies on gravity and stomach contractions to move food down. To prevent acid reflux, a partial fundoplication, wrapping part of the stomach around the esophagus, is sometimes done to act as a new barrier.
Tell me again, what's the new barrier? Because I was thinking if we can do without the sphincter, why do we even have it?
The new barrier is usually a partial fundoplication, where part of the stomach is wrapped around the lower esophagus to create a pressure zone that helps prevent reflux. We need the LES because, under normal conditions, it prevents stomach acid from flowing back into the esophagus. Without it, chronic acid reflux GERD can cause inflammation, ulcers, and even cancer over time.
One more time.
Got it. Want to wrap up with a quick
No.
LES normally prevents acid reflux by keeping stomach contents from flowing back up. After Heller myotomy, it's weakened or cut, so a partial fundoplication can be done to create a new pressure barrier. Without any barrier, chronic acid reflux could cause esophagitis, ulcers, or even Barrett's esophagus, a precancerous condition.
And
Did you want to dig into why?
no, I want to understand. Okay, we cut the LES. How is this barrier made out of? What is it made out of to prevent this acid reflux? What even is acid reflux?
The new barrier after a Heller myotomy is made from the stomach itself. A part of the stomach, usually the fundus, is wrapped around the lower esophagus in a partial fundoplication. This creates a pressure zone that helps prevent stomach acid from rising. Acid reflux happens when stomach acid flows back into the esophagus, irritating its lining. Normally, the LES keeps acid in the stomach, but if it's weak or cut, acid can escape, causing heartburn, inflammation, and damage over time.
Okay, so is it mechanical barrier?
Yes, the partial fundoplication acts as a mechanical barrier by increasing pressure at the lower esophagus. When the stomach contracts, it gently squeezes the wrapped portion, preventing acid from flowing back up. It's not a true sphincter, but it mimics one by using stomach tissue to create resistance.
Okay, that makes sense. Let's talk a bit now then about the another pathophysiology of esophageal motility, gastroesophageal reflux, GERD. Could we talk a bit about that?
Yes, gastroesophageal reflux disease, GERD, happens when the lower esophageal sphincter, LS, is weak or relaxes too often, allowing stomach acid to flow back into the esophagus. This causes heartburn, irritation, and inflammation, and over time, it can lead to complications like esophagitis, ulcers, or Barrett's esophagus, a precancerous condition.
So, can you explain a bit more? So, again, we have problem with the lower esophageal sphincter, and how will it? So, how will the tone be lost then?
The lower esophageal sphincter, LES, loses tone due to several factors, making it weak or too relaxed, allowing acid to reflux. This can happen because of 1. Obesity or high intra-abdominal pressure, which pushes against the LES. 2. Hiatal hernia, where part of the stomach moves above the diaphragm, reducing LES support. 3. Certain foods like caffeine, alcohol, and fatty meals, or medications like calcium channel blockers that relax the LES. 4. Nerve dysfunction, where vagal control or myenteric plexus regulation weakens. Over time, repeated acid exposure damages the esophagus, leading to GERD symptoms like heartburn and regurgitation.
Okay, and so can we use gastric acid secretion inhibitors then?
Yes, gastric acid secretion inhibitors like proton pump inhibitors, PPIs, e.g., omeprazole, and H2 receptor blockers, e.g., ranitidine, reduce acid production, helping to minimize damage and relieve symptoms in GERD. However, they don't fix the weak LES, so lifestyle changes or surgery, like fundoplication, may be needed for severe cases.
Okay, what about ulcers? You mentioned ulceration. What's that?
Ulceration refers to the formation of open sores in the esophagus, stomach, or intestines due to tissue damage. In GERD, constant acid exposure erodes the esophageal lining, leading to esophageal ulcers, which can cause pain, bleeding, and scarring. If left untreated, ulcers may lead to strictures, narrowing of the esophagus, or more severe complications.
Okay, I think this is a good point for us to go into the motor functions of the stomach. Could you explain to me how they store things and how they mix things? Hmm?
The stomach stores food by expanding when you eat, thanks to its rugae, folds, that stretch to hold more volume. The fundus and body act as the main storage areas, while the pylorus controls emptying into the small intestine. The stomach mixes food using peristaltic waves and churning movements from its three muscle layers, longitudinal, circular, and oblique. These contractions grind food into chyme, a semi-liquid mixture, and mix it with gastric juices, preparing it for digestion in the small intestine.
Okay, so talking about storage, what's fundus?
The fundus is the upper part of the stomach that primarily serves as a storage area for food before it gets mixed and digested. It can expand to accommodate a meal and helps regulate gastric pressure by allowing gases to accumulate before they are released, like burping.
I don't understand what the fundus is. So in storage, the fundus in the upper body of the stomach relax and they can accommodate the volume, the big volume of food. And why is this called receptive?
Yes, the fundus and upper body of the stomach relax to accommodate large volumes of food, a process called receptive relaxation. It's called receptive because this relaxation is triggered by swallowing. As food moves down the esophagus, the vagus nerve signals the stomach to relax and expand, preventing a sudden rise in pressure.
Okay. And what about smooth muscle relaxation? Does it have a correlation with storage? And what reflex controls it?
You have reached the message cap.
All right, let's break it down. The fundus is the upper part of the stomach, and its main job and motor function is storage. When food enters, the stomach has to make space for it, and that's where the vagovagal reflex comes in. This reflex is triggered when the stomach stretches. The vagus nerve sends signals both to and from the brainstem, causing the smooth muscle in the stomach to relax. This is called receptive relaxation because the stomach is receiving the food and making room for it without immediately contracting. This prevents pressure from building up too quickly and allows for a smooth transition into the mixing phase that happens later. It's a crucial step in digestion because without this relaxation, food Food would hit the stomach and create a lot of pressure,
Yeah, I
making the
get that.
process inefficient and
I,
uncomfortable.
I get that, but why specifically vagovagal reflex and fundus? Can you give me a phrase on the why for each one of them?
Sure, the vagovagal reflex is key because it allows the stomach to adjust its tone in response to incoming food, preventing excessive pressure buildup. The fundus is involved because it's the most compliant part of the stomach, meaning it can stretch easily to accommodate food without generating strong contractions.
Okay, got it. And so the antrum, what's the antrum?
I think you mean the antrum, which is the
Yeah.
lower part of the stomach. Its main job is mixing and grinding food before it moves into the small intestine. Unlike the fundus, which is all about storage, the antrum has strong muscular contractions that break food down into smaller pieces, making digestion more efficient. It's basically the stomach's mechanical processing unit before food gets released into the duodenum.
Okay, so what about mixing now? So we kind of already touched on like propulsion. So remind us. So we have slow peristaltic waves, and and how is this related to the pyloric antrum? And explain to us what the pyloric antrum is. We have the pyloric sphincter too. So talk to us about the etymology again of the pyloric.
All right, let's break this down. So, mixing in the stomach happens through slow peristaltic waves, which start in the mid-stomach and move toward the pyloric antrum. The pyloric antrum is basically the last part of the stomach before food moves into the small intestine. Unlike the fundus, which stores food, the antrum is muscular and propels food forward. But here's the catch. The pyloric sphincter, which is the gatekeeper between the stomach and the duodenum, doesn't let big chunks through. Because of this, when peristaltic waves push food toward the sphincter, most of it bounces back into the stomach, causing retropulsion, which is the key to effective mixing and grinding. Now, etymology time. Pyloric comes from the Greek word pyloros, meaning gatekeeper, which makes sense because the pyloric sphincter controls what gets released into the small intestine. It's like a selective exit door, ensuring only properly broken down food moves forward.
Okay, can we have one phrase on retropulsion again? And why? So is it cyclical, the propulsion, and then how does it correlate with chyme production?
Yeah, so retropulsion happens when peristaltic waves push food toward the pyloric sphincter. But because the sphincter is mostly closed, most of the food gets pushed back into the stomach instead of passing through. This back and forth motion keeps breaking food down into smaller particles, making it smoother and more digestible. Now, about the cyclical nature, yes, it's a continuous process. The stomach rhythmically contracts in waves, usually around 3 times per minute, pushing food forward, then forcing it back over and over. This keeps happening until the food particles are small enough to pass through the pyloric sphincter into the duodenum. As for time correlation, this process determines gastric emptying time. Larger, tougher foods stay in the stomach longer because they need more retropulsion cycles to break down. Liquids and very fine particles pass through much faster, so the stomach controls not just digestion, but also how quickly nutrients enter the small intestine, which is crucial for metabolism and energy balance.
I'm confused. Could you like tell me the same thing in 2 sentences? I'm really confused.
Sure, retropulsion happens when food is pushed toward the pyloric sphincter. But since the sphincter is mostly closed, the food bounces back into the stomach, creating a mixing and grinding cycle. This process repeats about 3 times per minute until the food is small enough to pass through, which controls how fast the stomach empties into the small intestine.
Hmm. Okay, this makes sense. So, okay, let's talk a bit about the regulation of gastric emptying and how it correlates to the overlying theme of motility of the GI tract.
All right. Gastric emptying is tightly regulated to make sure food leaves the stomach at the right pace for proper digestion and absorption. It's all about balance. Too fast and the small intestine gets overwhelmed, too slow and digestion backs up. This ties into the motility of the GI tract because the stomach isn't working alone. The rate of emptying depends on signals from both the stomach and the small intestine. The stomach pushes food forward with peristaltic waves, but the duodenum gives feedback. If it's already full or dealing with acidic or fatty food, it signals the stomach to slow down. Hormones like CCK, cholecystokinin, and secretin play a big role here. They tell the stomach to hold off on emptying when fat or acid levels are high, allowing more time for digestion. Meanwhile, the enteric nervous system adjusts muscle contractions across the whole GI tract to keep everything moving in sync. So, in the big picture of GI motility, gastric emptying is just one checkpoint in a coordinated process that ensures food moves efficiently but not too fast for proper digestion and absorption.
So, okay, I can't lie to you. I'm a bit confused. I'm getting one or two things. So okay, we have regulation of gastric emptying. So we have these peristaltic contractions that will force things through the pyloric sphincter. If you notice, I used the word things. I was going to use the word chyme. I'm not sure. What is chyme again? Chyme?
No worries. Chyme is the semi liquid mixture of partially digested food, stomach acid, and digestive enzymes. It's what food turns into after being broken down in the stomach. So, when we talk about gastric emptying, we're really talking about the stomach slowly pushing chyme through the pyloric sphincter into the duodenum. Duodenums. The rate at which this happens depends on the type of chyme. Um, Liquids empty fast, solids take longer, and fatty or acidic chyme slows things down even more because the small intestine needs more time to process it.
Okay, that kind of makes sense. So, can we talk a bit about the reflexes? So, do we have different types of reflexes? Like, do enterogastric and like I don't know. Does the ileum and the duodenum and the jejunum have different reflexes? What's going on?
Yes, the GI tract has several reflexes that help regulate motility and gastric emptying, and different sections, the duodenum, jejunum, and ileum, each play a role. First, the enterogastric reflex slows down gastric emptying. When the duodenum detects too much chyme, especially if it's acidic, fatty, or hypertonic, it signals the stomach to reduce peristalsis and tighten the pyloric sphincter. This gives the small intestine more time to digest and absorb nutrients before more chyme enters. Then we have the gastroileal reflex, which links the stomach and the ileum, the last part of the small intestine. When food enters the stomach, this reflex tells the ileum to increase motility and push any remaining contents into the colon. It's like making space for new incoming food. And there's the gastrocolic reflex, which makes the colon more active when the stomach is full. This is why eating can sometimes trigger the urge to go to the bathroom. So, yes, different parts of the small intestine and beyond have their own reflexes, but they all work together to coordinate digestion, absorption, and movement through the GI tract.
So if I have an increase in volume with these activate mechanoreceptors to like do things with the endocrine system and the enteric nervous system that will make us poop.
Exactly. When there's an increase in stomach volume, mechanoreceptors in the stomach wall detect the stretch. This activates both the enteric nervous system and the endocrine system, triggering different reflexes, including the gastrocolic reflex. This reflex signals the colon to increase motility, which can lead to a bowel movement. It's why some people feel the urge to go to the bathroom soon after eating a big meal. At the same time, the stretch receptors also activate hormones like gastrin, which stimulates stomach motility and helps move contents along the intestines. So it's a coordinated system. Your body prepares for incoming food by making space in the GI tract.
So what does enteric mean? Does it mean intestine?
Not quite. Enteric comes from the Greek word enteron, which means intestine. So when we talk about the enteric nervous system,
What did I say?
ENS, we're talking about the nervous system of the gut. It controls digestion, motility, and secretions independently of the brain and spinal cord, though it still communicates with them. It's
Wait,
sometimes called the second brain.
wait. I said enteric means intestine. Why did you say no? Also, why is the gut so important, and why do they say it affects our mood and stuff? If you could, like in 2 sentences, three perhaps answer briefly. Keep in mind, me and my audience don't have a big time span. Maybe we do have an attention span. I don't know. For interesting things, anyway. Excuse me.
All right. First, to go back to your original question, yes, enteric means intestine, and the enteric nervous system, ENS, is the independent nervous system of the gut. It controls digestion, motility, and secretions without needing direct input from the brain. Now, about the gut being the second brain, this is because the ENS has millions of neurons, almost as many as the spinal cord, and it can function independently of the central nervous system. It regulates digestion, communicates with the brain, and even influences our mood and emotions. This ties into mental health because the gut produces around 90% of the body's serotonin, a neurotransmitter that affects mood, sleep, and well being. The gut and brain communicate through the gut brain axis, meaning what happens in the gut can impact how we feel. Things like diet, gut bacteria, and digestion can influence stress, anxiety, and even depression. As for where the gut is, it includes everything from the esophagus to the colon. But when people talk about gut health, they're usually referring to the stomach, small intestine, and large intestine, where digestion and absorption happen.
Okay, so back up about the duodenum and the gastric emptying in general. So if we increase pyloric sphincter contraction and we decrease pyloric antrum contraction, this means that gastric emptying will decrease.
Yes, exactly. If the pyloric sphincter contracts more and the pyloric antrum contracts less, gastric emptying slows down. Here's why. The pyloric antrum is responsible for strong peristaltic waves that push chyme toward the pyloric sphincter. If these waves weaken, there's less force pushing food forward. At the same time, if the pyloric sphincter stays tight, it acts like a closed door, preventing chyme from passing into the duodenum. This combination keeps food in the stomach longer, allowing more time for digestion before it moves into the small intestine. This happens naturally when the duodenum senses too much acid, fat, or hypertonic chyme, triggering the enterogastric reflex, which tells the stomach to slow down emptying.
Okay, let's talk a bit about gastric motility pathophysiology more. So what about the dumping syndrome and gastroparesis? Tell us very, very briefly what these things are and how we can treat them.
All right. Dumping syndrome happens when the stomach empties too fast into the small intestine, usually after surgery like a gastric bypass. This causes symptoms like nausea, diarrhea, dizziness, and low blood sugar. Treatment includes eating smaller meals, avoiding high sugar foods, and sometimes medications that slow gastric emptying. Gastroparesis, on the other hand, is when the stomach empties too slowly due to weak motility, often caused by diabetes or nerve damage. This leads to bloating, nausea, and feeling full quickly. Treatment involves dietary changes like eating soft, low fat foods, prokinetic drugs to stimulate motility, and sometimes gastric electrical stimulation.
Okay, so why does dumping syndrome like cause cramping and diarrhea, and sweating and fainting? Because I understand, okay, we rapidly empty the gastric contents, but is it so? Okay, is it because we didn't absorb enough nutrients from the gastric contents to be like active and stuff?
Great question. The symptoms of dumping syndrome happen because of two main reasons. One, fluid shifts and osmotic effects. When chyme enters the small intestine too quickly, it's often hypertonic, high in sugars and undigested nutrients. This draws water rapidly into the intestine from surrounding tissues and blood vessels, causing cramping, bloating, and diarrhea. The sudden fluid loss from circulation can also lower blood pressure, leading to dizziness and fainting. 2. Hormonal and blood sugar effects. Because food is absorbed too quickly, there's a rapid rise in blood sugar, which triggers a surge of insulin. This can lead to reactive hypoglycemia, a sudden drop in blood sugar, making you feel weak, shaky, sweaty, and even faint. So, it's not just about losing nutrients, but also how the body reacts to the rapid entry of food into the small intestine, throwing off fluid balance and blood sugar regulation.
Okay, that makes sense. That makes sense. About so, but in one or two sentences, why cramping and diarrhea specifically, and how come the dietary changes help as a treatment?
Diarrhea happen because the small intestine pulls in too much water to dilute the hypertonic chyme, which stretches the intestine and triggers rapid movement, peristalsis, leading to discomfort and loose stools. Dietary changes help by slowing digestion, eating smaller meals, avoiding high sugar foods, and increasing fiber or protein prevents sudden fluid shifts and stabilizes blood sugar, reducing symptoms.
Okay, so let's go back to this because I'm a bit confused. So, in a sentence or two, explain to me cramping as a mechanism in diarrhea too.
Cramping and diarrhea happens because the excess water in the intestine overstimulates peristalsis, causing the intestinal muscles to contract more forcefully and irregularly. This rapid movement pushes contents through too quickly, leading to diarrhea and abdominal pain.
And so, okay, let's go to the etymology of gastroparesis. So, um, give us that, and then, okay, how exactly? So, yes, let's go to that.
The word gastroparesis comes from Greek roots gastro means stomach. Paresis means partial paralysis or weakness. So gastroparesis literally means partial paralysis of the stomach, which makes sense because it refers to the stomach muscles not moving food properly. Now, are you asking how gastroparesis happens physiologically?
Well, no, that okay. So paresis, what is paresis?
Paresis means partial weakness or reduced movement, usually due to nerve or muscle dysfunction. It's not complete paralysis, but a loss of strength or control. In gastroparesis, this means the stomach muscles are weak and don't contract properly to move food along.
So does gastroparesis have to do anything with gastric cancer or ulcers?
No, gastroparesis isn't directly related to gastric cancer or osteoporosis. Gastric cancer involves
Ulcer.
abnormal cell growth in the stomach, while gastroparesis is a nerve or muscle problem that slows stomach emptying. Osteoporosis is a bone condition, though severe malnutrition from gastroparesis could contribute to bone loss
Ulcers.
over time. That said, some conditions
I didn't say osteoporosis. I said ulcers. Ulcers. Also, why do prokinetic and anti-mimetic medication help with gastroparesis, and what even are they?
got it. Gastroparesis and gastric ulcers aren't directly related, but they can sometimes coexist. Ulcers are open sores in the stomach lining, often caused by excess acid or H. pylori infection, while gastroparesis is a stomach motility issue. However, delayed stomach emptying and gastroparesis can worsen acid reflux, which might contribute to ulcer formation. Now, about prokinetic and antiemetic medications. Prokinetics, like metoclopramide or erythromycin, help by stimulating stomach muscle contractions, moving food through the digestive tract more efficiently. Antiemetics, like ondansetron, reduce nausea and vomiting, which are
No,
common in gastroparesis due to delayed emptying. Together, they help manage symptoms by improving stomach movement and reducing discomfort.
I said anti memetic, is it the same? Antimemetic. Maybe I'm wrong. Is it instead called antiemetic? Yeah, I think I'm confused with the spelling, guys.
Nausea and vomiting, which are common in gastroparesis. Um, they don't speed up stomach emptying, but they make symptoms more manageable. Antimimetics, on the other hand, would mean something that opposes imitation or mimicry,
Okay,
which
okay. I got
isn't
confused
relevant
then.
here.
Yes.
So we're definitely talking about antiemetics for gastroparesis.
Okay, okay. Sorry. Okay. Alrighty. Let's continue a bit about the intestinal motility. So, in the small intestine, what's going on in terms of motility there? We have nutrient absorption. What else?
In the small intestine, motility is all about moving and mixing food to help with digestion and absorption. Besides nutrient absorption, key motility functions include segmentation, rhythmic contractions that mix food with digestive enzymes and maximize contact with the intestinal walls for better absorption. Peristalsis, wave-like contractions that push food forward toward the large intestine. Migrating motor complex, MMC, a cleanup wave that happens between meals to clear out undigested material and bacteria. All of this ensures proper digestion, prevents bacterial overgrowth, and prepares waste for elimination. Anything specific you want to dig into?
Oh, this is good. So basically, you have propulsion of chyme, or whatever it's called, hemos, I assume, in Greek, via peristaltic waves, and we also have the segmentation that we talked before. So, is the segment? Why do we do segmentation? Does it have to do with the exposure to like absorptive epithelium or something?
Yeah, exactly. Segmentation is all about maximizing the exposure of chyme, the partially digested food, to the absorptive epithelium of the small intestine. The back and forth contractions mix the chyme thoroughly, ensuring that nutrients come into close contact with the villi and microvilli, which absorb them. It also slows down movement, preventing food from rushing through too quickly. This gives digestive enzymes and transporters more time to work. Without segmentation, absorption would be way less efficient.
Can you summarize in a sentence or two? And it's good. You mentioned microvilli. Also, we have mucosal villi and we have plicae circulares. So explain obviously the etymology of all of these, and then explain to me why we need this surface area that these things give to us.
Sure. Segmentation helps mix chyme and slow its movement, ensuring better contact with the absorptive surface of the small intestine for efficient nutrient absorption. Now, for the etymology, microvilli equals small, villi equals finger-like projections are tiny extensions on individual cells. Mucosal villi, villus equals shaggy hair, are larger finger-like projections of the intestinal lining. Plicae circulares, plicae equals folds, circulares equals circular, are circular folds in the small intestine that increase surface area. We need all this extra surface area because it massively boosts absorption. More folds, villi, and microvilli mean more contact points for nutrients to be absorbed efficiently into the bloodstream. Without them, digestion wouldn't be nearly as effective.
Can you summarize in a sentence or so I'm confused?
Sure. Microvilli, villi, and plicae circulares increase surface area in the small intestine, helping absorb more nutrients efficiently.
Why?
Because more surface area means more contact between digested nutrients and the absorptive cells.
And what
This
are
allows for better nutrient uptake into the
okay,
bloodstream,
I got that
making
part.
digestion more efficient. Without these structures, absorption would
Okay,
be
okay. So explain to me what plicae circulares is then, and what's the difference between mucosal villi and microvilli, and what even are they? What's the etymology in a sentence or two, please?
plicae circulares are large circular folds in the small intestine that slow down food movement and increase surface area. Mucosal villi are finger-like projections of the intestinal lining, while microvilli are tiny projections on the surface of individual cells within the villi. Etymology: plecae circularis, circular folds, plecae folds, circularis, circular. Villi equals shaggy hair, refers to their finger-like shape. Microvilli equals small villi, micro equals small, villi equals projections. All three structures work together to maximize nutrient absorption.
Can we summarize this in a sentence or two, perhaps?
Plecae circularis are large circular folds. Mucosal villi are finger-like projections on these folds. And microvilli are tiny extensions on individual cells. Together, they greatly increase surface area for better nutrient absorption.
Oh, dude. I don't know. I'm sorry. I'll put timestamps. Can you explain it again?
Yeah, it all stacks up, fully and microvilli, just to maximize absorption. Pretty cool, right?
So, what's the structural differences between plica circularis, mucosal villi, and microvilli?
The structural differences are all about size and location. Plica circularis are the largest structures, circular folds made of both mucosa and submucosa. They slow down chyme and increase surface area. Mucosal villi are smaller finger-like projections that sit on top of these folds made of just the mucosa. They contain blood vessels and lacteals for nutrient absorption. Microvilli are the tiniest microscopic extensions on the surface of epithelial cells lining the villi. They form the brush border, which has enzymes to help break down nutrients further. Basically, it's a layered system. Plicae circularis, villi, microvilli, each level increasing absorption efficiency.
Mm, Um, okay. So mucosal
You're
villi
welcome.
are they?
Glad we
Okay, so mucosal villi are in the mucus, and then microvilli are where? Where is their location? I don't understand.
Here's how they're located. Plicae circulares are large circular folds found throughout the inner lining of the small intestine. Mucosal villi are finger-like projections on these folds, extending from the mucosal layer. Microvilli are tiny hair-like extensions on the surface of the epithelial cells that line the villi. So it's a nested structure. Plicae circulares contain mucosal villi, and mucosal villi have cells covered with microvilli.
What?
Is there something specific you'd like me to clarify or explain further about their locations?
Perhaps all of it. So, okay, now it makes more sense. Let's talk a bit about excitation and inhibition. This has to do like with hormonal regulation of motility. How does this work? We've already mentioned secretin and motilin. How do these work in terms of motility in the small intestine?
Great question. So motilin, for example, stimulates the migrating motor complex, which helps move things through the small intestine between meals. On the other hand, secretin mainly regulates pH by stimulating bicarbonate secretion, but it can also modulate motility by slowing gastric emptying. It's all about balance.
Okay, so why is motilin in charge of excitation and like insulin too, and why is glucagon and secretin in charge of inhibition?
So motilin acts during fasting to trigger those strong contractions that clear the stomach and small intestine. Insulin can promote motility by influencing glucose uptake and energy availability. On the flip side, glucagon and secretin slow things down. Glucagon raises blood sugar and diverts energy from digestion, while secretin helps control acidity, which can slow gastric motility.
Okay, let's talk a bit about propulsive peristaltic reflexes now. And we should connect them to obviously the overlying theme of motility of the GI tract.
Absolutely. Propulsive peristaltic reflexes are essential for moving contents through the GI tract. They involve coordinated muscle contractions that push food forward. This reflex is triggered by stretch receptors in the gut lining. It ties into motility by ensuring contents move efficiently from one section to the next.
Mhm. Interesting. Okay. So, w- okay. Um, Can we talk a bit about the migrating motor complex? What's this, and does it connect?
Sure, the migrating motor complex, or MMC, is like a housekeeping wave that sweeps through the stomach and small intestine between meals. It happens every 90 minutes or so and clears out leftover food and bacteria. This keeps things moving and prepares the gut for the next meal.
Okay, so it's basically a series of peristaltic contractions.
Exactly. It's a series of strong, rhythmic contractions that move from the stomach through the small intestine, ensuring everything is cleaned out.
And why is it called migrating motor complex and it isn't called peristaltic contractions?
Good question. It's called the migrating motor complex because it migrates in a repetitive cycle and isn't continuous like normal peristaltic waves. It's a distinct pattern of motility that migrates through the gut during fasting, hence the name.
Hmm, interesting. Okay, so um, let's talk a bit about the final stages of digestion then in the large intestine.
Sure, in the large intestine, water and electrolytes are absorbed, and any remaining material is compacted into stool. Beneficial bacteria in the colon also help ferment undigested carbs, producing gases and vitamins like K and B12. Everything then moves toward elimination.
Hmm,
It's a pretty
interesting.
efficient process. Glad you're finding it interesting. Anything else about digestion you'd like to know?
Yeah, so how does water absorption in the ascending and transverse colon work? This reminds me of the nephron.
Great comparison. In the ascending and transverse colon, water absorption is driven by osmotic gradients, similar to the nephron. Sodium and other electrolytes are absorbed first, creating a gradient that pulls water out of the chyme. This helps compact the waste as it moves along.
Okay. And so why is absorption of water in the sorry guys, ascending and transverse colon but feces formation is the descending colon?
That's because the ascending and transverse colon are primarily for absorbing water and electrolytes, reducing the volume of the chyme. By the time it reaches the descending colon, most water has been absorbed, so the focus shifts to forming and storing feces until elimination.
Yeah, but I don't understand why. Can we go into the like uh, benefits of having the ascending and transverse colon doing water absorption, whereas I guess feces formation is the last step, so it's the descending, right?
Exactly. The ascending and transverse colon absorb water to reclaim fluids, which is crucial for hydration and electrolyte balance. This also helps compact the waste. By the time it reaches the descending colon, most water is gone, so it can solidify into feces for elimination.
Okay, so talk to me about the commensal microbiome then and how it relates to the final stages of digestion in the large intestine.
Sure, the commensal microbiome in the large intestine plays a key role in breaking down undigested fibers, producing beneficial short-chain fatty acids, and synthesizing vitamins like K and B12. These processes help maintain gut health and support the final stages of digestion.
And why is it called commensal microbiome?
It's called commensal because these microbes live in a mutually beneficial relationship with us. They get a place to live and nutrients from our food, while we benefit from their digestive help and immune support.
I see. And what's the biome part?
Biome refers to the community of living organisms in a specific environment. So, the commensal microbiome is the collection of beneficial microbes in our gut, contributing to our overall health.
Hmm, interesting. Okay. And what about the okay, let's talk a bit about the motility in the large intestine. So we have yeah.
Sure, motility in the large intestine is slower than in the small intestine. It involves haustral contractions, which mix contents, and mass movements that push material toward the rectum. This ensures efficient absorption and compaction.
So what's hostral bulges?
Haustral bulges or haustra are the pouches formed by the muscle tone of the colon.
And how do they connect to what we just talked about?
Haustral contractions help mix and slowly move the contents, aiding in water absorption. This slower movement ensures thorough absorption and compaction of waste, preparing it for eventual elimination.
Hmm. Okay,
Pretty
but so what's the name hostel? Where does it come from?
Haustral comes from the Latin word hostra, meaning bucket or sack. It refers to the pouch-like segments of the colon that help in mixing and absorbing water from waste.
Okay, so we have this longitudinal muscle then, and it's thickened in the large intestine, and then it contracts to create hostile bulges.
Exactly. That thickened band of longitudinal muscle, called the tenei coli, contracts to create the haustral bulges, which segment and move the contents slowly.
And what about hostile churning?
Haustral churning is when these haustra contract to mix the contents, helping in water absorption and moving them slowly toward the rectum. It's a crucial part of forming solid waste.
Okay, okay, let's talk a bit about the propulsion though, so via the mass movement. So we have peristaltic contraction, they force contacts in. I just want to understand. Make sure they force the contacts into the sigmoid colon and the rectum, and this can happen up to like 3 times a day when we poop, basically.
Exactly. Mass movements are strong, wave-like contractions that push contents through the colon into the rectum. They usually occur a few times a day, often triggered by eating, to move waste toward elimination.
Okay. Lastly, before we end this crazy long podcast, let's talk about the defecation reflex.
Sure, the defecation reflex starts when the rectum is stretched by feces. This triggers signals to the brain, creating the urge to go. The internal anal sphincter relaxes, and when you're ready, the external sphincter releases to allow for elimination.
And why is it called defecation?
Defecation comes from the Latin word defecare, meaning to purify or cleanse. It refers to the body's process of expelling solid waste, completing the digestive cycle.
Hmm. Okay. And okay, so I think I got that. So basically, we have these feces, basically, and they activate stretch receptor, and then yeah, so the enteric nervous system is activated.
Exactly. When feces enter the rectum, they stretch the rectal walls, activating stretch receptors. This triggers the enteric nervous system and signals the need for defecation.
Got it. Good, good. And do we have also like parasympathetic innervation too, or no?
Yes, we do. The parasympathetic nervous system plays a big role by promoting peristalsis and relaxing the internal anal sphincter to facilitate defecation.
Okay, and then how does relaxation of the internal, like an external anal sphincter happen?
The internal anal sphincter relaxes involuntarily when the rectum is distended. The external anal sphincter, on the other hand, is under voluntary control, allowing you to decide when to relax it for defecation.
I see, I see. Okay, guys, you're not gonna believe this. This is the longest podcast, obviously, but we officially finished with it. I'm quite happy. I think you noticed it in the tone of my voice too. So yeah, let's make a summary. So could you summarize to us the motility of the GI tract?
Absolutely. GI motility involves coordinated muscle contractions to move food through the digestive tract. In the esophagus, peristalsis moves food to the stomach. The stomach mixes and propels chyme into the small intestine, where segmentation and peristalsis continue nutrient absorption. The large intestine uses haustral contractions and mass movements to absorb water and form stool, leading to defecation. Each step is crucial for efficient digestion.
Okay, and I think I'm gonna start doing this as a normal thing on the end of each podcast, guys. I'm gonna ask you, ChatGPT, a few questions. Firstly, if we were in a magic machine and forgot everything we just listened to in this podcast, what would be one thing worth remembering about our topic?
If there's one key takeaway, it's that GI motility is essential for moving food through the digestive system. ensuring nutrients are absorbed and waste is efficiently eliminated. It's a coordinated process that keeps everything running smoothly.
Okay. And what would be a practical application in terms of knowledge to use this in real life? And I want you to remember, take it from different perspectives, from a medical student's perspective, a doctor's perspective, just a person that's interested in learning, or a patient.
From a medical student's perspective, understanding GI motility helps in diagnosing and treating digestive disorders. For a doctor, it's crucial for managing patient care, especially in conditions like IBS. For someone just interested in learning, it's a fascinating insight into how our bodies work. For a patient, it helps in understanding symptoms and the importance of a healthy diet and lifestyle.
Interesting. Okay, and now if I link it to philosophy and the big problems that we have in the world and big questions, for instance, scientific questions or philosophical ones like consciousness and ethics, and problems in the world like war and environmental problems and stuff like that. And I know this sounds a bit patronizing, guys, but I want us to, even the most random subject, to try to at least in some way connect this to what we're talking about at the end of the podcast. What would you say the connection is, ChatGPT?
That's an interesting challenge. GI motility, like many bodily functions, showcases the intricate balance and interconnectedness inherent in nature. This can be linked to philosophical concepts of harmony and balance, similar to how ecosystems function. It also touches on the importance of understanding complex systems, which is crucial in addressing global issues like environmental sustainability. Just as the body requires balance for health, our world requires balance and understanding to tackle big challenges.
Interesting. Can you go a bit deeper into that?
Sure. Just like the GI tract relies on coordinated effort for motility, addressing global issues requires coordinated global action. The concept of balance and digestion reflects a broader need for balance in ecosystems and societies. Understanding the intricate workings of one system can inspire us to approach complex global challenges with similar care, recognizing the importance of each part in contributing to the whole.
Wow. This rings nice bells in regard to reductionism and holism.
Absolutely. It's a perfect example of how reductionist and holistic approaches can complement each other. By understanding individual components, we gain insights, but appreciating the system as a whole is essential for true understanding and effective solutions.
Great. Okay. So, ChatGPT, um, do you want to say anything before we close the podcast?
Just that, it was a pleasure joining you. Keep exploring and connecting ideas. And remember, understanding even the smallest processes can offer valuable insights into the bigger picture. Thanks for having me.
Yeah, that's amazing detail about details themselves, because sometimes I feel I get lost in them. So, well, you've heard it from the AI itself, and I want to remind you guys, maybe I'm wrong, correct me if I'm wrong, but ChatGPT is a prediction of what we would say as a whole, the next most predictable thing we'd say. So it's the wisdom of the crowd, it's us, and we are speaking to ourselves through it, helping each other. So having said that, I hope this was a nice episode, guys, and see you at the next one. Love you.